Okay, so how these different matrices are useful for giving the specific score when we are doing the sequence alignment. So sequence alignment, this is basically called the heart of bioinformatical analysis, because without doing this particular alignment of any kind of sequence and we are getting the information out of it, we cannot able to progress for any kind of further work towards the structural and the functional analysis of a particular protein or related to a particular gene. Okay, so what we basically try to do here, after aligning the either protein sequence, otherwise nucleotide sequence, we try to find out what kind of similarities are there between those sequences and what type of relatedness also present on those particular sequences, and based on that we try to conclude something. What basically we can able to conclude, like if we found the similarities and relatedness are present among those particular sequences, we can tell genes and genetic element, what kind of genes are present in that particular sequence and what kind of genetical element also present in that particular sequence. We can able to understand from that. Then it will help us to creating the molecular phylogeny, that is creating the ultimate phylogenetical tree, and from there we can able to understand what kind of relations are there between all those organisms. And of course it is very much useful nowadays for study the comparative genomics. Okay, entire genome to genome comparison, and we can able to understand what way the genome have, if that particular gene or entire genome have come for evolution process, and what kind of modifications are there in the gene composition level. All the aspect we can come to know by doing the comparative genomics, and of course that is depending on the sequence alignment. And finally we can do the homology modeling to when we are going to build the structure of the protein, when we are going to know that what kind of folding will be there of the particular RNA and what kind of folding should be present also in the DNA level. So all those activity we can able to find out if we are doing the sequence alignment activity. That's why I have mentioned the sequence alignment is considered as the heart of bioinformatical analysis. So what actually this sequence alignment, how we can define this sequence alignment? This is sequence alignment is nothing but arranging the protein, otherwise DNA or the RNA sequence one after another, and after arranging them one after another, a position wise we try to find out what kind of similarities are there, and based on the similarity or dissimilarity we try to extrapolate the functional, structural, and the evolutionary relationship between the sequences. So alignment, it is nothing but arranging the sequence, how we are arranging, and after arranging, how we are getting the information from those arranged sequence. So that is the activity of the sequence alignment process basically we need to do. And every sequence alignment, after doing, we provide a certain kind of score. And based on the score, if it is a more positive score, we can tell the sequences are very close to each other. If we see that more negative score, then we can tell the sequences are more away from each other with respect to the phylogenetical tree. So sequence alignment, you can see here, this is the arranging of the nucleotide sequence. This is one sequence, and below this is the another sequence. And this way, arranging the sequence and try to match each alphabet to alphabet is main prime of this sequence alignment process. So this is with respect to the nucleotide. I'll be coming this part. This some dotted lines are present here. This is called the gap. So why this gap should be introduced and what is the function of this gap, I'll be coming in my lecture. So this is the activity called the sequence alignment with respect to the nucleotide. And similarly, with respect to the protein also, we can able to do the similar kind of activity. So you can look at here, this is the protein sequence alignment. So this is one particular protein from human, then chimpanzee, then mouse, then rat and cow, etc., etc. So we have aligned one after another, and all the protein is the histone H1 protein and almost having 120 to 180 residues. So once we are putting them one after another, we can see, or we can look this every line. So if you look at this particular first position of this all sequence, you can see here all are K. Then second line also present all are K. So like that, if we move, if for every position of every line, we try to find out what kind of alphabets are present, and based on those particular alphabets, we can conclude like these are the conservative part, or this is the non-conservative part, or this is the semi-conservative part, all those aspects. So beginning I mentioned, until unless we are doing this particular sequence alignment, we may not able to understand what kind of function this particular protein is having, what kind of structure this protein is going to make, all those aspects. So it is very important for us. If I'm doing a research, I got a particular protein sequence. Now what I can do with the protein sequence? I need to go for next this sequence alignment process. And by doing this sequence alignment, I'll be getting some information that my protein must be having similarity with so and so protein, and so and so protein, how this structure and the function it is having, most probably my protein also going to have similar kind of structure and the function. So this is the activity. It is possible if we are doing this kind of alignment process. And you can look at here, we are providing here for alignment purposes many sequences one after another. So this activity is called the multiple sequence alignment. Whereas if you look at here only two sequences I have put it together and we are aligning. So this process is called the pairwise sequence alignment process. So multiple sequence alignment and pairwise sequence alignment both are having huge importance in the bioinformatical field. So first I'll be talking about the pairwise sequence alignment, and next I'll be coming to the in details about the multiple sequence alignment process and why multiple sequence alignment is very much important. All those aspects I'll be explaining. So if you look at the DNA or the RNA, that is made up of completely nucleotides. So nucleotide, either it will be having ATGC, or sometimes in RNA we can get the uracil. Okay, so with if we are aligning the any DNA sequence or any particular RNA sequence, so only this 4 alphabet ATGC, what kind of differences and what kind of similarities are there, we try to find out that. But if we come to the protein level, so proteins, as we know that our biological system can able to encode the 20 different kind of amino acid, and for that there are having 64 different codons, and out of that 3 are the nonsense codon, and apart from that all other codons can able to encode the 20 different valuable proteins, means amino acid. And for some amino acids there is having 3 or 2 different type of alternative codon also. But anyhow, there are proteins are made up of with the 20 different kind of amino acid. So if we align the proteins, we try to get the idea about this particular amino acid, whatever the present in the respective position of that proteins. But proteins, as per the biochemistry rule, the protein can be designated with either capital alphabet, like capital A for alanine, capital R for arginine, capital N for asparagine, capital F for phenylalanine, etc. Or we can able to abbreviate by the 3 different alphabet, like ALA, A is capital L, then small l and small a, that is the alanine, like capital PRO, proline, etc. So in the bioinformatical field, or whatever the sequence basically we need we get, so all the sequence basically it will be designated with the single capital alphabet. So single capital alphabet, that will be useful for designating the amino acid. So you can look at here, all are having single capital alphabet. So these are all indicating the specific amino acids. So what is the goal of this particular sequence alignment process? So major goal of the sequence alignment process to find out the common ancestor among those sequences. Okay, so if we do this kind of multiple sequence alignment, we try to find out or we try to get the idea who is the common ancestor. That means if we look at the cow, and if we consider cow is the ancestor, or if we consider the rat is the ancestor, then from rat, after evolution, how this particular histone protein have came to the cow, and then successively to the mouse, chimpanzee, and ultimately to the human. So this is the concept of finding the common ancestor. So based on the common ancestor, we can create the phylogenetical tree. But for if you are doing the pairwise sequence alignment, they are trying to find out that what kind of mutations basically happening or basically present among those particular sequences, and based on the mutation, how evolutions are happening. So mutation finding is the major prime important parameter if we comparing the DNA or the RNA sequences. So if we look at the DNA sequence, as per the basic concept of the genetics or molecular biology, we know that deletion, substitution, transversion, transitions, these are the various processes are there for happening the mutations. It could be point mutations, it could be multiple position mutations. So many things can happen. So in the DNA level, basically we can see the major after aligning the sequences, we get that these two important terms, that is the transitions and the transversion kind of mutations. And some position, of course, we can see the insertion also there, and some position also you can found some kind of deletions of nucleotide also present. But major idea is try to get the positions where the transitions and transversions are happening. And transitions, so you can found in the more frequently than the transversion in the sequence level. And the transition and the transversion, how it is happening, that is going to find out by these nucleotide sequence alignment. Whereas if we talk about the protein sequence, so proteins, the 20 amino acids, whatever is there, the 20 amino acids are having different type of properties, a different type of side chain. And based on the properties and based on the side chain, whether hydrophobic, hydrophilic, the many criteria are there. So we have grouped this 20 amino acid again into the 3 or 4 different subgroups. For example, if the subgroups, if we talk here, suppose valine, arginine, lysine, it is present in one group, then histidine, phenylalanine, etc. is present in the another group. So these subgroups in terms of what kind of physicochemical parameter or properties those amino acids are having based on that. So when we are aligning the protein sequence, if we found one position is present K, and next in the same position if it is present A, then K and A, if it is belonging to the same group, then we can give a particular score. And if K and A completely belonging to the two different group, then we can provide some kind of negative score for them. So we need to, you have to understand that what basis we are going to give this kind of positive score or negative score. And ultimately, we will try to understand these two terms, that is whether these sequence or these particular alphabets are synonymous or these alphabets are non-synonymous. For the DNA level, ATGC, whether purine, purine, and pyrimidine, pyrimidine, we used to tell these are the synonymous. And if it is purine and some position pyrimidine has come, we used to tell this is the non-synonymous. But for the protein, if when we are aligning, we need to have to be very careful for telling this is the synonymous sequence or synonymous amino acid, or this is the non-synonymous amino acid. So for protein, we need to go through proper way. But for the DNA, it is very easy, or the nucleotide sequence, when you are aligning, it will be very easy to extrapolate the informations. So this is the, with respect to the DNA. So if you look at here, this is one sequence, this is another sequence, we have put it together. And this particular highlight part, it is telling upper part position is present T, and the same position down sequence, it is also present the T. Next, same second position also T, third position also it is T. Third position again G, down it is present the C. So G and C, it is not matching. Whether TT it is matching, then GG is matching, AA is matching. So like that. So we need to try to find out where exactly this matching alphabets are present. Where there is no matching alphabet, that means this position is called the mismatching. So based on the matching and the mismatching, we need to provide certain kind of score. And you can look at here in this alignment, there is having certain score like expected 3 minus 136. Then out of 291 amino alphabet, 284 are found they have identical. Identical means in case of nucleotide, the T and T, G and G, C and C, A and A, wherever is matching. So total number of matching, they have tried to find out the percentage. Then how many gaps? So gaps is indicating wherever the dash positions will be there, that is called the gaps. So look at here, here is one particular gap is there. So what is the importance of this gap? I'm going to tell it now. So this is the way, basically, if you are aligning the two different nucleotide sequence, we try to get such kind of score, and by looking this score, we need to conclude that these sequences are perfectly matching, and this sequences, whether it is the homologous sequence, non-homologous sequence, etc. So sequence homology versus sequence similarity, this is the two terms we need to understand. who are both with respect to the protein sequence as well as your nucleotide sequence. So similarity, it is telling basically the likeliness or percentage of identity between the two sequences. How much these two sequences are identical. To finding out the percentage is called the similarity, and whereas the homology refers to the share of ancestry. That means who is the ancestor of this particular compared sequence. So that is finding out is basically in the terms of homology we can explain. And similarity means again the sharing of statistically significant number of bases or amino acids, and how many positions we are getting match. So with respect to the match, when we are talking about the score, there will be having lot of statistical applications, statistical procedure. So based on the statistical procedure, we have to signify that, and based on the significant value only we can tell these two sequences are very much similar. So this statistical parameter, it is completely applicable when we are talking about the similarity. Whereas for homology, two sequences are homologous if they are derived from the common ancestor. So there we are not looking for this kind of statistical approach. Just by looking, yes, it is same A, same TT, same GG, same. So somehow we are telling these are somehow to be a homologous sequence. So similarity does not imply the homology, whereas homology usually imply the similarity. So look at this example. So if we look this particular two sequence, so this is one sequence, this is another sequence. If we look the alignment, V and V is matching, I and I is matching, again VV is matching. So only L and V, this particular position is not matching. So we can tell with respect to the similarity, these sequences are 90% similar. Fine. But at the same sequence, if we look for telling the homology, so you can see the V and T, this is the difference, and the I and S, this is different, V and Y different, only AA is matching, but rest of the position it is not matching. So we can tell this is the 10% similar, but we never tell these two exact alignment things are very perfect. And we never tell, when we are talking the homology, we never tell these two sequences are, suppose this sequence is the originator of this sequence, or this sequence is originator of this sequence. So that idea we may not able to get from here. Only we can able to tell with respect to the alphabet to alphabet matching, how many similarities are there, how many, and what is the non-similarity residues percentage wise present among these two particular sequence. So this is one aspect. And second aspect is the finding the sequence similarity versus the sequence identity. So sequence similarity and sequence identity, with this example you will come to know. So sequence similarity and sequence identity with respect to the nucleotide, it is the common. Like it is a both the side of a particular coin, head or tail, whatever it may be, but it is the part of that coin itself. So that is why I'm telling with respect to the DNA and RNA, this is the same meaning. But if we come to the protein sequence, and if you look at this particular table, I was telling just a few minutes back that the amino acid can be grouped into the various way, like AVPFM. These are the one particular group, and that is basically small hydrophobic, including the aromatic amino acid also is present. D, that is the acidic, RK is the basic, and this is the group, it is hydroxyl sulfidyl amine group, and also glycine also present in this group, like that. So in the when we analyze this particular protein, so there will be having particular color annotation, especially this color annotation we can get in the multiple sequence alignment process. So you need to understand this red color indicating which particular amino acid, blue color indicating which amino acid, and based on that we can come to the certain kind of conclusions. But this group is very important with respect to the protein structure and the function. So if we talk about the identity and the similarity with respect to the protein sequence, so identity is indicating the percentage of exact matches found between the two aligned sequence, whereas similarity it is telling percentage of aligned residues that share the similar characteristics. So similar characteristics, so how many times this particular group 1 amino acid got replaced by the amino acid, that is telling the you're sharing the similar kind of physical and the physicochemical property. So you can understand identity and similarity with respect to the protein sequence, it is completely different. Whether the for nucleic acid sequence, it is the very same thing, like opposite side of both the coin. So you'll able to understand this similarity, identity, and homology with this particular example. So here, this is the comparison between the two sequence of TFR1, that is the transferrin receptor protein 1. So both the transferrin receptor protein have taken from the mouse, and one has taken from the human. So when we compare these two particular protein sequence, we got this kind of alignment. And if you look in this alignment, so as we are comparing two similar protein taken from the two different organisms, so this is indicating this is the homologous sequence. Whereas similarity, if you are able to understand, look at this particular part. You can see that V is there, down also V, upper T is there, here is H. This plus is indicating similar group of amino acid. Whereas G is there, here is below is also G. So this GG is perfectly matching. So wherever the plus is indicating, this is indicating the similar group of amino acid, and exactly similar amino acid, whatever is there, a same alphabet also written here. So this part implies the similarity segment of this particular protein. Whereas identity, if you look at, if you are able to understand, you can see here exactly all the alphabets are matching in this particular positions below of these two particular sequence. So this is identity, similarity, and homology with respect to the protein. It is very important for us to understand, because DNA level, that will be similarity and identity, whatever I'm talking, that with respect to the either adenine, otherwise guanine, or better than we can tell that is the what type of nucleotide sequence based on that. So it is not a difficult task with respect to the DNA or RNA, but it is a difficult task to understand with respect to the protein. So this kind of similarity and identity, that is having a particular equation. So we are putting this equation, and finally we are getting this kind of similarity and identity related all such kind of score. You can see here, this is the some kind of score is mentioned, and somewhere the gaps are there. I already mentioned the gaps, I'll be explaining afterwards. So this is the activity with respect to the protein we are getting, and by using this kind of formula, basically this formula to be set it into the software. When we are writing the algorithm, we are putting this particular equation mediated algorithm there. So software, we have to provide the sequence, software will analyze the sequence, and immediately within few seconds or few minutes, it is going to exhibit such kind of aligned result with the proper score. And the score will be coming because of this particular type of mathematical equation. Now, you need to understand that what is the method of this alignment, which particular method I supposed to follow, and what way I supposed to get the information after doing this alignment. So method of alignment, basically there are having two important method of this alignment. So first you look at here, this is one particular sequence and red is another sequence. So these two sequence, when are having hypothetically similar type of nucleotide or exactly similar number of exactly same number of amino acid, then we can able to compare them from end to end. Like this N terminal end to C terminal end, and this protein also N terminal end to the C terminal end. So when you are aligning this way and try to get the information related to the entire sequence, so that is the process called the global alignment. Okay, globally means universally. So that from universal, that what has come the global. So entire sequence we are giving the preference. We are trying to do the alignment and try to get the information. So global alignment. Whereas if you look at here, suppose this is my particular sequence, I want to know about this particular sequence, and this sequence already is present in the database, or somehow somebody else is having this sequence. I'm going to compare these two sequence. So here my sequence, wherever 30 to 40% will be matching in between this particular sequence. So I'll be trying to match my sequence from there itself, and I'll be putting this sequence, and I'll be extrapolating, or I'll be stretching this particular sequence towards the C terminal end. So here only the partial part or a particular segmented part, it is starting for this kind of alignment. So it is called the local alignment. Locally, wherever it is matching, we are trying to put my sequence with the particular comparable sequence and try to compare between these two. So there are two important methods for aligning the sequence. One is called global alignment and second one is the local alignment. So a global alignment, we are assuming, as I mentioned, that both the sequences are having that equal length. But truly speaking, it is not possible. So suppose your sequence is having hardly 300 amino acid or 300 nucleotide, to whom we are going to compare might be it is having 600 or 700 or 1000 nucleotide or amino acid. So the global alignment, though the main assumption is here, both the sequences will be having the similar type of length, but sometimes it is not possible to get exactly. So this is the process of global alignment. I'll be giving the example. So this is the process of global alignment. So this is one particular sequence with whom I am going to compare my sequence. So query sequence is my sequence and target sequence with whom I am going to match. So this is the process. I am going to find from 5' end what particular sequence is present in the target sequence, and my sequence 5' end which particular alphabet is present. I am putting them one after another and try to extrapolate my sequence. And when I'm trying to extrapolating, just a few minutes back I mentioned, so target sequence might have 1000 different nucleotide and your sequence might have only 500 or 600 nucleotides. So in that case, when you are going to match it, so that time some position you need to provide the gap and you need to extend your this particular sequence. And when you are extending this particular sequence and try to reach towards the 3' end of the target sequence, so that time this gap we have to provide. Otherwise, you may not able to cover to the entire 1000 alphabet which is present in the target sequence. So basically, software it is doing the algorithm how we have set it here. It is based on the algorithm. This particular activity is going on. So basically, global alignment, end-to-end encryption, end-to-end alignment. After doing the alignment, if you try to extrapolate the information, that is called the global alignment. Whereas a local alignment, here the assumption is both the sequence definitely does not have the similar type of alphabet or the similar type of lengthwise similar number of alphabet. So if it is a nucleotide, same number of ATGC will be not be there in the both the sequence, or if it is amino acid, so both the sequence will not be having similar number of amino acid. So in that case, if you are going to compare, so that process will be the local alignment. So in the local alignment, what we are doing, so this is your sequence. In this sequence, without providing any kind of majorly gap, I repeat, without providing any kind majorly gap. So what we used to do, this is your sequence. This sequence from starting from 5' end. So 40% alphabet wherever it will be matching with the target sequence. It could be in the exactly from starting to second, third positions. It could be in between any positions wherever it may be. So you need to start aligning that from that particular position, and without gap, you need to put your sequence, and we need to find out the relations or find out the alignment activity with respect to the local positions of target sequence as well as the query sequence. So this is called the local alignment. And similarly, we can do this kind of alignment for the protein. This example, whatever I provided, that is with related to the nucleotide. And this is the example with respect to the amino acid, that is the protein. So global alignment, this is the one sequence, and this is the your query sequence. So query sequence is having very short number of amino acids, whereas your target is having more number of amino acids. In that case, you need to provide the gap, you need to stretch your sequence. Whereas local alignment, here also we need to provide the gap. If the gap is very much necessary, then only, and the gap should not be too many number. Okay, so hardly we prefer there is having in this algorithm, so there is having a particular segment. That segment is completely fixing that total gap should be there in the local alignment, not more than 12. Not more than 15 number. Okay, so if you're aligning, the gap should not be 15 number, but for global alignment, the gap should be n numbers. Fine. So this is the activity, local alignment and global alignment is happening. So I'm just showing a particular practical example. So pairwise sequence alignment, for that you can open the Google, you can type the pairwise sequence alignment, this particular word, you will be getting this kind of Google page, and you can click on this pairwise sequence alignment of EMBL EBI, European Molecular Biology Lab, European Bioinformatics Institute. So you just click over this particular hyperlink, immediately this page will open in front of you. So you can see here global alignment, local alignment, two different terms are mentioned. So for the global alignment, basically that algorithm is called the Needleman-Wunsch algorithm. So you need to click on this particular needle, and immediately we can will be reaching to this particular page where you can able to paste your sequence and you can continue your sequence alignment activity. And if you're ready to do the local alignment, then this is the, you can see local alignment is written. So local alignment, that is the Waterman algorithm it is called. So you need to click on this emboss and immediately you'll be reaching to the local alignment page. So first I'm talking about the global alignment. So for the global alignment, you need to click on the needle. And once you are clicking on this hyperlink part of the needle, you are going to get, this is the home page called emboss needle home page. So this is for pairwise sequence alignment, and you can see here protein is written, and here one arrow will be there. If you're clicking on this arrow, so there will be written protein or nucleic acid. So if you are working with the protein, you just leave it as a protein, and you need to paste your protein sequence here in the FASTA format. So this is the FASTA format, one protein sequence I have taken, and this is the greater than symbol followed by the definition line, and then alphabets are there. This is the major form of the FASTA format. So you are providing one sequence here, and immediately there will be having another dialog box where you can put the another sequence. So what you are going to do, you are going to compare with this sequence and this sequence. So both the sequence we are going to compare, and first sequence whatever you have put it here, so say for example this is your target sequence, and second box whatever the sequence you are putting, suppose this is your query sequence. Fine. So these two sequences you are going to compare. So after pasting both the sequence in the FASTA format, so you need to provide your mail ID, because it may take some time to giving your result. So the result hyperlink will be directly coming to your mail. So if you are providing the mail ID, and after providing the mail ID, just you need to click below the submit button will be there. You need to click on the submit button. So once you're clicking on the submit button, it will take some time. The hyperlink result will be appearing to your mail. And if you're clicking, if you're opening your mail and clicking on this hyperlink, immediately you'll be reaching to this particular page. And this is called the result page. Okay, result page with respect to the your this particular emboss needleman algorithm method. Fine. So this is the global alignment method. So in this method, first it will be written, these are the two sequence related information, that means how many alphabets are there and what kind of alphabets, means what kind of amino acids are present in your sequence. So all those aspects will be written first, and below if you scroll down, you can see this is the alignment. Fine. And this alignment, this is the particular part is indicating gap, and somewhere this is the dash is there, somewhere it will be there one small dot. So all these are indicating whether it is the physicochemically similar group of amino acid or it is completely different group of amino acid. And exactly the same amino acids are matching, like P with the P is matching, then your Y is matching with the Y, then this one particular stroke will be there. And this is the result basically we can get with the your global alignment by using the needleman algorithm. So emboss needle, this particular algorithm if you are using, so we are going to get this kind of result. With the same sequence, if we follow the local alignment, that means I mentioned here. So first when you are clicking on this particular homepage of EMBL EBI, so here is having local alignment. So in the local alignment, this water emboss part you need to click off. So once you are clicking it, you are going to get the local alignment, this particular activity page. Okay, so pairwise sequence alignment, this of the local alignment it is called. So here also, if you are putting the same protein sequence in both the box, and after that if you are providing your mail ID and you are submitting it, so the result again it will be coming to your mail, and from that hyperlink if you are clicking, you are going to get, this is the result page. You just look at here, both the results with the same proteins are having different meaning, right? So this different meaning is based on the algorithm, how it has written. So this algorithm is deciding this score and this alignment pattern.
Okay, same protein, you just see here with this 2 line itself, they are finishing everything. Fine. So exactly local matching wherever they are getting, based on that they are providing the result. Whereas this sequence, when you are doing that multiple your global alignment process, so we can see how many lines are there, and based on that how you are getting the result. Fine. So you are, I hope you able to understand that pairwise alignment we can do with the either with the global alignment process, otherwise you by local alignment process. So coming back, coming back to the again to the alignment algorithmic process. So I hope you got understand. So global alignment and local alignment with the same sequence, if you are performing in the 2 different cases, you are going to get the 2 different kind of score and 2 different kind of result. So when you are doing the research work, which particular method you have followed and which algorithm you are following, definitely you need to mention. Otherwise, your result will not be accepted and it will be a not authenticated one. So you have to remember, so which method of alignment you are selecting and which algorithm you are using. So both the things you need to clearly need to mention in your thesis. So now come to the alignment algorithm. So I'm every time telling this particular activity result is based on the algorithm. So all the softwares we are using, the software how it is following the instruction. It is following the instruction based on the algorithms, and algorithms are nothing but the language of the computer software. By using the specific language, we are giving the instruction, the software in such a way so that it will follow one after another various step, and finally once they are completing all the step, the result is going to appear in front of your monitor. So you need to understand what kind of algorithms are there behind this particular global alignment or the local alignment. So initially when this algorithms were designing, so that time first concept to have been created, that is called the dot matrix method or dot matrix concept. Here in the dot matrix concept, what is to do, we need to take the two sequence we are going to compare. So both the sequence you need to keep in the X and Y axis, and alphabet to alphabet we are going to match. So look, suppose this is one graph paper, you have created the 2 axes and you have put it, this is one sequence. Suppose this is your target sequence and this is your query sequence. So both the sequence, it could be of similar lengthwise similar, or it may not similar length, doesn't matter. So you are putting both the sequence in the 2 different axes, and wherever the alphabets are matching, you are just going to put a dot. Fine. So here N and M is not matching, you are not giving anything. N and N is matching, putting a dot. Again N and N is matching, putting a dot. Like that, each word you need to consider, and based on that you are going to put a dot. And finally, after getting all the dot, so you need to go through from diagonally, and you need to join from down to top diagonally all these particular dots. So and freehand, if you're joining this dot, you are getting, suppose, this kind of one line. So these lines and these dots are having huge meanings. And as by putting the dot, wherever matching is there, by putting the dot, we are creating this kind of matrix, and that algorithm is based on this. So it is called the dot matrix algorithm. So now this is, I have given just one example. So various way you can, if the sequence length is too much, this is the with respect to the nucleotide. So we can get the various position, this kind of dot, and after that all the possible dot diagonally, if it is able to join, we can join it and we can get, these are the various line. Okay, and this is the main diagonal line, and both the half of this line is having again another few parallel line. So all these lines are having a particular significance. Somewhere we can get this kind of line also. Here is a straight line, then here is a vertical line, and here again the diagonal line we can get. So these sometimes we can get this kind of line also. So what actually these lines are indicating? So this is the gist form of your dot matrix. So after getting the line, suppose if you are getting perfectly diagonal line, so this is called the perfect match between the 2 sequence. If you are getting perfect diagonal line and both the half is having the certain percentage of this particular diagonal line, So this is indicating this short stretch of line indicating this is the repetitive sequence, and this particular part is the matching sequence. Whereas if you're getting completely this kind of cross, so that is indicating both the sequence are palindromic sequence. Like that, if you're getting one diagonal line and partially crossing line, so this is called the partial palindromic sequence. That means only this particular part and this particular part is having palindromic sequence. Whereas if you're getting, these are the particular region, so many dots you are getting in this part, and if you are imagine, it could be possible to make a particular square box. So this particular part is going to indicate the microsatellitic portions, even mini satellite, the homologous, indel sequence, all those aspects with this particular dot matrix we can able to get. So this dot matrix was initial consideration when we are going to compare between the protein-protein sequence or between the nucleotide-nucleotide sequence. So this is the outcome of the dot matrix. But for if you wanted to use the dot matrix, there will be dot matcher. So it is from EMBL, UBI. They are taking care. So just this URL if you are using, and if you are providing your both the sequence, so this kind of dot matrix it will be generating and it will be coming in front of your monitor. So if you are having this particular idea, so these lines indicating what, you can easily get the idea. So what type of similarity and matching or mismatching etc. is there between these two sequences. Apart from this dot matcher, there is having dot helix, matrix plot etc. So all can going to give the similar kind of result only. But now only dot matcher is available freely, but dot helix and the matrix plot, they are not providing the free accessions, or free access is not possible. So only the dot matcher it is possible. So now next algorithm, as the dot matrix are having the major drawback. What is the drawback? Drawback is you just look at here this sequence. So okay, how many alphabet is supposed to put into your x-axis and how many alphabet supposed to you are going to put to your y-axis? That is a big question mark, because if your sequence contains for example 5,000 alphabet, both the sequence might have together around 1 billion of nucleotides or 1 billion of base pair. So in that case, how long or how many base alphabet is supposed to put in this particular graph paper? So it is a big question mark, right? So there is having a drawback. This is the major drawback of this particular dot matrix. So ultimately the dot matrix concept now we are not considering, and slowly we have evolved another kind of matrix that is called the dynamics programming matrix. So dynamic programming matrix, it is another name is the Needleman-Wunsch algorithmic matrix. So this Needleman and Wunsch together, this particular matrix they have developed, and all kind of software freely available, whatever is there, we are following the this pairwise sequence alignment method. There's this Needleman-Wunsch algorithm itself is going to perform, and finally we are getting the result. In the Needleman-Wunsch algorithm, they have proposed first time that we need to give certain kind of score. Wherever the matching is there, we need to give the score. Wherever mismatching is there, there also need to give the some kind of negative score. And if we need to provide the gap, I mentioned sometimes you need to extrapolate our sequence so that we are creating the gaps. So if you need to provide the gap, so there also we need to give some kind of score. So actually with this particular scoring pattern and the scoring the idea, if you are comparing any kind of sequence, so we are going to get very perfect result, and it will be easy for us to understand this sequence, how it is perfect or similar, how it is matching, whether it is homologous, etc., etc. So the scoring concept first introduced in this case in this dynamic matrix. So in the dynamic matrix x, y, z, three different coordinators we are considering, and these three coordinators we need to take care when we are providing the particular score. For example, if we are plotting, this is one sequence, this is another sequence. So wherever matching is there for the first line, so A and A is matching, we are putting 1, AT is not matching 0, wherever is not matching, it is the 0. So now if we come to the second line, so G and A, it is not matching 0, but G and T, it's supposed to be 0. Now, as I mentioned, when we are putting the 0 or any number, you need to consider both the x, y, and z, three different axes. So here G and T is supposed to be 0, but if you look at the three axes, so here one maximum number is there. So instead of putting 0, you need to place 1. Like that, if you move to the second part, so second again G and T should be 0, but if you look at the your XYZ axis as 1 is here, you need to place 1 again here. So like that, if you move to the third box, so G and G, it is matching. So for matching means it will be 1. Now, before putting 1, again you have to look to the XYZ coordinates, and in the this particular box, it is having 1. So 1 plus 1 together 2. So here you need to put the 2. So like that, it is putting the 2, and this way that matrix are supposed to be filled up. And after filling up this matrix, you need to move to the diagonally. So diagonally, last alphabet is the 4. You need to move to the diagonally next 3. Then it is coming 1, and immediately after 1, if you are getting here 1 1 1 or any similar number of alphabet, you need to go horizontally, and where this number is ending, again from there you need to go diagonally. So this is the with respect to these two particular sequence. And now by looking this diagonal line and by joining this number, you are going to give the alignment. So here 4, so it is indicating the C and C is matching. 3, it is indicating G and G is matching. And next, wherever this particular stage horizontal line you are getting, that is indicating first one is the gap or mismatch. And next, how many numbers it will be repeating, that will be the gap. So look at here, T and G is matching, putting together, that is indicating mismatch. Then it is having the your gap, and finally again A and A is matching, so it is putting together. So this way the gapping concept have been introduced perfect way, and this dynamic matrix perfectly explaining why this gap is so much important when we are doing this final alignment. That means when the software is going to provide the final result in front of you based on this particular dynamic matrix concept, we are getting this kind of gapping part. Fine. So next, this dynamic concept of once it is over, then as I mentioned, this Needleman Wunsch, they have tried to put it some kind of score. Okay. So initially it was 1 and 0, and if it is matching 1 plus 1, how I have explained, it was going on smoothly like that. So after that, they have mentioned that this is here only the very few alphabet I have taken in the sequence. If the sequence alphabets are becoming too much lengthier, then this gapping number also sometimes will be increased. And sometimes some positions you need to provide the at least 5, 6, 7, sometimes 10 different this kind of gap. So they have later on they have tried to produce this gap penalty. Okay, this gap penalty concept they have introduced. Okay, so wherever the gap is creating, we need to provide some kind of negative marking, and all the matching score, mismatching score, negative marking, all will be summating together. Then what score will be getting? That score will be ultimate score of these two alignment. Fine. So this gapping score, when they introduce, again they have considered that gap extension and the gap penalty and gap extension. So what is the already I mentioned Needleman Wunsch, all this I have shown. So gap extension and gap penalty, what is the concept of it? If you look at here in this two particular sequence, so this is one sequence, this is another sequence. So here is having only single gap. So now if it matching, if we consider, you can see here total match, how many is there? So for each match +1, and if mismatch is there, if you putting the -1, and for each gap, if you are putting the score -2, then with respect to this particular two sequence, we are getting the score like -1. Fine. Now, when we are introducing the affine gap penalty or extension gap penalty, that concept, so these two sequence, again, if we are considering, just look at here, this is having a single gap. So the single gap means we are going to give it -2. Whereas if we look at here, here is having two simultaneous gap. So first gap, like below T, whatever the gap is creating, that is called the opening gap. And next below A, this gap is called the extension gap. So opening gap should have, as per this particular algorithm or this particular theory, this opening gap should have maximum negative marks, and the extending gap will be having very minimum negative marks. So for example, if T below the T, whatever the gap is creating, if we are taking it -12, below A, whatever the gap is creating, we can give the score like -1. So -12 and -1 we are considering here, and here only the, if you are giving -12, we are considering only -12. So what is the difference between these two? Here there is a no proper affine gap penalty concept, but here is having affine gap penalty concept. Ultimately, if you are getting many stretch of this gap, we are gap opening and gap extension, two different kind of penalty as per this algorithm need to provide, and finally we can get the score. So you can understand this dynamic programming method why logically is very much famous, and why people are going to accept this particular kind of matrix. So I talked about just now about the algorithm. Now I'll be coming to the scoring matrix. Okay, so scoring matrix, it is basically two different matrix we follow in the bioinformatics, all kind of software. One is called the PAM matrix, and second one is the blossom matrix. So blossom matrix is much more useful with respect to the protein, because the already I mentioned protein different grouping of amino acids are there, whether the substitutions and matching and mismatchings are happening among those group or in between this group. All those aspects we are giving consideration, taking consideration here, and based on that blossom matrix we are creating. And PAM matrix, it is going to consider only the point accepted mutation pattern. So you will understand what is the difference between these two matrices. So first matrix is the substitution matrix. So substitution matrix, if you look at for nucleotide level, so there we can, once we are aligning the sequence, these are the three important things we are going to get over there. One is either identical nucleotide, either AA, TT, GG, CC is matching. Otherwise, transitions pair. One is top is A, below is G. So this is the transition, or it will be vice versa. Of top is G, below is A, or T is their top, below is C, etc. So transition pair, and the transversion pair like AT or TG or TA, GT. This kind of situation we can get. So for nucleotide, only this particular table, whatever I mentioned, only this table we need to consider, and that's why the DNA or the nucleotide related comparison and getting score is very easy. Fine, but for the protein, it is very crucial to understand that about the each and every amino acid, and this amino acid, what kind of properties there, which particular group it belongs, each and everything. So this grouping for amino acid is completely depending on the physicochemical property, and substitution among the similar residues are likely to be preserved and essential, and it is going to give the proper function and the structural aspect of the protein. So if you are taking 2-3 different protein, so each protein, that particular position, which amino acid is there, that important positions of, if it is important positions for that protein, you will be able to understand why these amino acids are exclusively present in that particular positions. So there is having the statistical tool. That statistical tool is going to prove that essentiality of that amino acid in that particular positions. So if we are, this is the different amino acid group, non-polar side chain and the polar, etc. Even among them are having some are acidic, basic, etc., etc. I'm not going in detail. So generally, phenylalanine, tyrosine, and tryptophan, and all these amino acids sharing the similar type of aromatic ring. So chemical structure wise and function wise of all these three amino acids are almost same, and that's why one can replace to the in the protein to the other, and it is very frequent, and there is a no particular function wise and the structural wise any deformities or any differentiation we can get between the proteins. So these are the as belonging to the same group and same property they are having. So whatever the proteins we can see in this universe, so these positions of this particular position of a protein, if it is phenylalanine is there, similar type of protein, same position, tyrosine if it is there, it is obviously nature has accepted. Like if you see the arginine, lysine, and the histidine, these are the basic residues, and they are having high probability among the being substituted with each other. So one can substitute with each other very frequently. We can get in the protein like aspartic acid, glutamic acid, asparagine, glutamine. These are all having acidic property, and suppose they can able to substitute with one to other that also you can found in the very frequently among the protein. Like that, hydrophobic may not able to frequently replaced by the hydrophilic amino acid, because hydrophobic amino acid in the protein structure, if you see, that will be present always to the core part of the protein, whereas hydrophilic protein amino acid should be present exterior or the outer surface of the protein. Similarly, serine, threonine, and your cysteine, the amino acid which can make the disulfide bond, these are very, very much crucial to be present in the active part or active core site of that particular protein. So all the protein universally it is accepted, it is found this particular amino acids are present exactly active site part of that, but all group of protein. That means nature is not going to accept that particular amino acid should be replaced from that particular positions. So this idea we are having now, based on this idea, actually first time Margaret Dayoff proposed that particular amino acid scoring matrix, and that matrix name as per her, so the matrix name is the point accepted or point percent accepted mutation matrix or percent accepted mutation matrix, or we also call them as a Dayoff amino acid substitution matrix. So Margaret Dayoff, she was basically physics persons. She used to work on the biophysical aspect, and first time she only proposed. So this way we can score the amino acids, and based on her scoring pattern, this bioinformatics subject start developing. So it was 1978, and the journal was Atlas. In that, she has published first time her this paper, and Blossom matrix also is depending on this particular PAM matrix considerations. So Blossom is the modified one, but the fundamental basis of the Blossom matrix is the PAM matrix. So Margaret Dayoff, she has selected 71 families of protein, and then 71 of these families, again it was belonging to the 34 super families, and she has created the matrix. Even PAM matrix we can have the different variation, PAM 250, PAM 10, PAM 100. These are, so PAM 250 scoring matrix, it is telling 250 substitution is possible among the 100 amino acid. PAM 10, 10 substitution is possible out of 100 amino acid. Like that, PAM 250, PAM 10, etc. different variations have been created. But how Margaret Dayoff created? Margaret Dayoff has taken, these are the few important group of proteins. So immunoglobulin, kappa chain region, then casein protein, epidermal growth factor, serum, albumin, etc., etc. And she has aligned all this particular protein, and then she has created particular PAM matrix process. So mathematically, this particular process is there, and this is not the correct platform to explain the mathematics how it is happening, but it is the fundamental basis and completely depending on the logod or the logod score. So in this matrix, whatever the score you can see, this is the final matrix. If we follow this kind of mathematical procedure, so definitely we are going to get this kind of values. So this will be the fraction value, this value to be multiplied sometimes with the 2, sometimes it can be multiplied by the 5, and that's way we are going to get this kind of completely complete number. Fine. So this is the matrix we are following. So once we are using the PAM matrix, we are comparing between the two protein. So the software will be following this particular logic and this particular protocol, this particular table, and based on that, the matching, mismatching, etc., it will be considered, and final score will be presented in front of you. So this is the very important kind of fundamental matrix which was created by the Margaret Dayoff. And the next matrix, which is called as the Blossom matrix. So I'll be telling briefly about the Blossom matrix too. So the Blossom matrix, it is called the block substitution matrix. So this is a block, you can understand we are creating some block or some group, and what kind of substitution happening between them. So that is going to consider here. And again, here also log score value we are calculating by the mathematical formula. And finally, we are going to get a matrix. This is called the matrix called the Blossom matrix. So this is one particular block. This is NDEQ is another block, HRK third block, MILV is another block, and FYW is another block. So this Blossom matrix is called the 2020 matrix, because in the x-axis we are keeping the 20 alphabet of amino acid, y-axis also keeping the 20 alphabet of amino acids, and we try to find out that C can be replaced with the C, what is the chances are there, C to be replaced by the S, what kind of chances are there, So the chance factor, the probability factor, we are basically calculating here, and that way we have created this kind of blossom matrix. So with respect to the blossom matrix, we can score whatever the example I have mentioned just in the beginning, like your alignment, okay, pairwise alignment or global alignment or local alignment. So all their alignment, there will be written what kind of matrix they have used to provide your result. And when you are doing this kind of alignment for your research purposes, definitely you need to write what kind of algorithm you have used and which particular matrix you have used, and based on that you got this kind of score. All those things you need to mention in your thesis or in your report. So this is the blossom matrix, and in each block, so what is the substitution values, what kind of probability is there, based on that these matrix have been created. So PAM and blossom matrix having some similarity and dissimilarity. And if we are doing this kind of alignment, basically it is very much useful for multiple sequence alignment. So here, if we consider we have aligned the 10 different sequence, and if we look at one particular line among this particular sequence, and this line is going to indicate you one particular block. Repeat, this particular line is going to indicate this is one particular block. Fine. And first created blossom matrix was blossom 62. So if we consider this particular alphabet, so how many time A is present here, how many time S is present here, and what is the chance that S should be present in this particular positions, and what is the chance to be present all this alphabet A in the similar positions. So everything we are calculating with respect to the probability, and having this is the mathematical part. Again, I'm not going to talk, this is not the correct platform. So with all these particular things we can consider, and finally with respect to this one single line, we can able to create the blossom pattern or the blossom matrix mediated result. So this blossom matrix is very important, and PAM matrix rather than PAM, blossom is going to useful for multiple sequence alignment, and from there to creating the phylogenetical tree. So next the BLAST and the FASTA, whatever the process are there, for each process we are using this particular blossom 62 matrix to getting certain kind of score. So fundamentally this particular matrix is depending on, again, the day of whatever the proposal has given. So it is completely based on that particular proposal, and this proposal have bit modified here and ultimately created this block substitution matrix or the blossom matrix. So I'm going to tell the next part, that is database similarity searching. So you have understand that right now whatever I have talked, that is based on the two different sequence. So once both the sequence of your choice, right? The example whatever I mentioned here, so both the sequence is your own choice. Fine. So this is the sequence, it is your choice, and this is the sequence also, it is your choice. And between these two group of protein sequence, you are going to compare and you are going to get the result. Now I am going to talk, concept is same, but it is having a different kind of approach, and that approach is called the database similarity searching approach. So last lecture I was talking about the database and how database is very important. So nucleotide database, protein database, different databases are there, and different group of databases are also present. Among the protein, different classification group is there, and among the nucleotide also having different variations are there. So here by doing this pairwise alignment, how we can compare your protein with the database, whatever the similar kind of protein or similar type of nucleic acids are there with them. So the big difference is here your query sequence will be your own, but target sequence you will not able to know. Just by using this particular software or this method, you are just providing the query sequence. Based on the query sequence, databases, whatever the similar sequences are there, your query sequence will be matched with each database sequence. And of course, there will be having pairwise sequence alignment method. They'll be matching it, and with respect to the each database sequence, they are going to give the alignment result. So that is the difference between the database similarity searching method and general pairwise sequence alignment method. But I am repeating, database similarity searching method, that is having two methods, BLAST and FASTA, both are depending on the pairwise sequence alignment process only. That means the scoring pattern and the alignmenting process, it could be the similar. Only the algorithmic part is different both for the BLAST as well as the FASTA. So BLAST, this is very, very, very much useful protocol or the procedure for any kind of bioinformatical research. When you start doing our journey in the bioinformatics, immediately first you need to understand what is the database, how to extrapolate the information from the database, how to download the sequence from the database, and then what you can do with the sequence. You can do the if you can do with the sequence. First thing is the BLAST, otherwise FASTA. If whatever the sequence you are having, you need to understand with whom our databases, whatever the sequences are present, with whom it is going to perfectly match, what way it is matching, and what way I'm going to tell this is perfectly match or mismatch. So for that, BLAST is very, very much needed, and BLAST is very much essential. So BLAST stands for Basic Local Alignment Search Tool, and FASTA stands for FAST-AL. Okay, FAST, FAST all. That from there, FASTA is what has created. So among this database similarity searching method, FASTA was the first created method. In this FASTA method, there was having certain kind of slowness criteria. That means it can take the lot of time to means analysis and give the result, and researcher will not be having so much time to wait for that. So that's why BLAST is quite faster than FASTA method. And nowadays we are using worldwide this BLAST method. Now, of course, BLAST is also having various kind of variations. So first I'll be talking about the BLAST, and both BLAST and FAST are coming out the database similarity searching protocol or the process. So database similarity searching, when we are doing, few important criteria should be remembered. That is the sensitivity, selectivity, and the speed. Sensitivity, that means what kind of sequence I'm providing, whether it is a protein sequence, nucleotide sequence, that sensitivity must have this particular software. And based on that, if it is a nucleotide sequence, it is going to knock the nucleotide databases, and it is going to compare with the nucleotide databases whatever the nucleotide sequences are there, and based on that it is going to give the score. The score whatever is going to get, that is called the true positive score. Second important thing, that is the selectivity. So what sequence you have provided, you have given the for example hemoglobin sequence, and the database it is going to search with the actinin, tubulin, and XYZ different protein. So that should not be happen. So as you are providing the hemoglobin sequence, databases hemoglobin sequences whatever will be there, first you need to compare with that, then subsidiarily it can be able to compare with all other kind of protein also. So that is called the selectivity. What way it is going to select, and what way it is going to compare with the databases sequences. And third important point is the speed. Just now I was telling, no researcher having so much time and so much patience to be waited for 1 day, 2 days, 3 days, 5 days. So we always want to, after doing the activity, immediately we want the result, so that next step we can carry forward. So these are the three important parameters for all kind of database similarity searching. It is important. And for the FASTA, this speed is the factor. But for the BLAST, there is no problem with the sensitivity, selectivity, as well as the speed. So BLAST is the basic local alignment search tool, and the BLAST have created or BLAST is basically well known as a particular term, that is the Google of biological research. Okay, so whenever you used to do the BLAST, whenever we are having a sequence, everybody used to tell, do the BLAST, do the BLAST. What is the BLAST? BLAST is, if you are opening the Google page, you are typing the BLAST, immediately the BLAST page is opening and you are using it. That's why it is very famous by this particular term also, Google of biological research. And it is, BLAST is following the heuristic algorithm. That means word to word it is going to match. That is the meaning of heuristic conditions. So it is going to provide the heuristic algorithm. And just in the year of 1990, before that FASTA was very famous, but in the year of 1990 from NCBI itself, Alstuch and Warren Garif and the David Miller, these are the three major people, along with the David J. Lipman of the National Institute of the Health, NIH from USA. They have suited together, make this particular algorithm, and finally they have published their paper in the Journal of Molecular Biology 1990. And within 2 months, it got citation around 75,000. and now you just, if you imagine, it will be a few billions also. So it is so much famous. Okay, whoever is working on this particular sequence comparison process, they have to use the BLAST, and immediately they are going to give the reference of this particular paper. That's why the reference is coming, citation is coming too much, and based on the citation you can understand how means important content present in this particular paper. That's why everybody is following that paper. So this BLAST basically how they are doing. So BLAST, I mentioned, it is the heuristic method of algorithm, and here also looking for the make the local alignment process. And once they're having the local alignment method, they are considering a few keywords. Okay, so what the method, as I mentioned, heuristic approach. Heuristic approach is indicating what by what comparison. So here it there in this BLAST algorithm, it is the what by what comparison protocols will be there. So how what by what protocol will be there? Here one short example. Suppose this is your sequence GLKFA you have provided for analyzing the BLAST. So initially first three alphabet is going to consider, that is the GLK, then LKF, then KFA. Like that, three different words have been created with this. So GLK is the first word, then LKF is the second word, and then KFA is the third word. So BLAST algorithm or the BLAST method, this way selecting the few words, and these words they are extending right hand side and left hand side, and it is going to compare with the your target sequence, and wherever the maximum score is coming, that score there it is going to indicate this is the perfect alignment with respect to that particular sequence. And that way the BLAST is going to give very faster and accurate result. And as I mentioned, the sequence to be moved right hand and the left hand directions, that means the neighboring sequence whatever is there, the neighboring sequence also is coming under the picture, and that way it is 100% guaranteed it is going to give the perfect result. And here when we are comparing, that should be in this algorithm a particular threshold value. The threshold value is called the T value. So when we are moving towards the right hand and the left hand side of this particular alphabet or the particular sequence, if this particular right hand side, once we are moving, the score whatever is getting, if it is crossing the threshold value, then we can expect this is the much more the reliable alignment part. And if the threshold value it is not crossing, we can understand this is the mismatch part, and this part we supposed to not consider here. And by doing this, definitely, as I mentioned, I am talking the threshold score and some value. So for giving the value or particular digit or number, Blossom 62 matrix to be applied here. So for all BLAST, Blossom 62 is going to apply, and here threshold determining process is very, very important. And without this threshold part, if you not able to previously determining this threshold part and the threshold value, the BLAST algorithm is not going to work out. So there is having a definite threshold value, and with respect to the threshold value, entire thing we are comparing. So I'm in this algorithm, one more thing it is there, that is to finding out the low complexity region or LCR region. LCR region is stating the suppose some protein and the nucleotide sequence having too much repetitive part. Okay, A, T, 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 G, 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 and like that, or ATGC, 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 like that, different repetitive part if it is there. So that repetitive part in this BLAST algorithm may create some kind of ambiguity. So the first step for starting this kind of algorithm to find out the low complexity region from the provided structure, and BLAST is going to remove that particular low complexity region first from the structure. And when they are removing the low complexity region part for a protein, the output whatever will be coming, there will be written X, and N will be written for the nucleic acid. So look at here, this is the alignment result once we are getting. So here X, 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 X something is written. So this X are indicating these are the particular positions of the query sequence was having lot of this repetitive sequence. That means that was having lot of low complexity regions. So BLAST during this particular alignment and during the scoring, so they have removed this particular positions for considering the scoring process. Okay, so that's why it will be written first. So first thing of this algorithm, try to find out the low complexity region, then determine this K letter, that means word. Okay, so generally for the BLAST, this K word, here example I have given is a K is equal to 3, but generally for protein, this K will be usually 11, and for DNA for DNA it will be like 4, right? So for the for the protein it will be 3, and for DNA it will be the, or any nucleic acid, it will be the 11. Fine. So if you look at here, this is your query sequence. So if it is the protein, so first 3 alphabet, then second 3 alphabet, then third 3 alphabet, like that each word will be created, and this word will be compared with the threshold value of the respective positions, and if it is crossing the threshold value, this particular word is going to match over there, otherwise that particular part will be discarded. So this is the concept. Now why this is the 3 for amino acid and this 11 for the DNA or the RNA? Because 20 amino acids were there. Out of 23 always we are considering, because based on the Weibull hypothesis and based on the codon approach. Okay, 3 alphabet together going to make a single amino acid. So that's why out of 20, these 3 we have considering here. And for DNA and RNA, out of 4, this 11 we are considering. Just remember, out of 4, 11 we are considering. So ATGC, this 4 alphabet will be there in the DNA. So at a stage ATGC, GC, CGC, like starting whatever the 11 alphabets are there, that 11 alphabet will be considered, and that will be compared with the that respective target sequence with that. And if it is crossing the threshold value, then that particular position, that alignment is perfect, and we are considering that. Then next 11 alphabet we are taking, and that way we are crossing the software is covering the entire length of your query sequence as well as the target sequence, and finally they are going to project the final score. Okay, and here score for Blossom 62 matrix to be used, and in the Blossom 62 matrix, in this particular case of the BLAST, suppose +5 will be for match, mismatch is -4, and +2 as a -3 will be there for your nucleotide. So nucleotide for matching +2, for mismatching -3, whereas for protein +5 will be for matching, mismatching will be -4. And if it is a gap extension and the gap penalty, so gap penalty will be the, of course, -4, gap extension will be -1. So this way ultimately the entire thing is going to cover, and finally we are going to get, as I was telling, this alphabet what we are considering, there will be a threshold score. So ultimately we try to find out if 3 alphabet we are considering, this 3 alphabet exactly this position how much score is giving. Fine. So if you look at here, this particular positions, so left hand side is having these 2 alphabet, suppose right hand side is having these 2 alphabet. So with respect to these 3 PEG, we are getting the score here as a plus 23. So this is called a high speculated projected score. Okay, HSP, or sometimes it is also called high scoring segmented pair score. So always try to find out this high segmented pair score, and here the threshold value, if it is crossing, so definitely we are accepting this is the particular positions, perfect alignment is there, and this should this is the conditions will be reflected in front of your monitor when the result output is coming. So this there are having, by this way BLAST is working, and there are having different variation in the BLAST, BLAST 10, BLAST P, BLAST X, T BLAST 10, T BLAST X, etc. So BLAST 10 for DNA or nucleic acid, here query sequence to be DNA, we need to get the output in the terms of DNA itself. BLAST P, protein sequence we are providing, output should be protein. BLAST X, here DNA we are providing, but protein sequence we are getting. This BLAST X is very important to searching the mutational pattern of the protein, and then or mutational pattern of the DNA. Both the cases, this BLAST X is very, very important. And T BLAST 10, we are providing the protein, but the output will be coming in terms of the DNA. And the T BLAST X, here the DNA we are DNA should be query sequence, and from the DNA what kind of translational product may come, and what kind of variations should be there in the translational product. So that kind of approach we can use with the help of T BLAST X. So generally the BLAST we need to provide, this is the particular algorithm, www.ncbi.nlmn.nih.gov/BLAST. So we can open the NCBI, from there also we can go the BLAST. Otherwise in the Google directly we can search the BLAST, and we can able to reach to the BLAST page. I'll be showing that particular activity, how generally we are using the BLAST. And this BLAST, when I'm beginning itself, I mentioned the BLAST and FASTA. It is useful for comparing the sequence or it is useful for actually comparing the sequence with the database, whatever the sequences are there. So definitely when you are doing the blast, if I wanted to compare my sequence with the uniport, whatever the nucleotide sequence is there, only those uniport containing nucleotide sequence I want to compare, or else I want to compare with the your scope or catch or the different software, different databases with related to the protein I need to compare, I can select that. Otherwise, directly, if you're not giving that particular path, I am having only options to compare my sequence with this particular protein. It is going to compare with all the nucleotide sequence or all the protein sequence connected with the NCBI. It is going to compare with the each and everybody. Fine, and that way it is going to give the result. So nucleic acid related, we are having non-redundant database like GenBank, EMBL, DDJB, EST, etc. is connected. Chromosomal database, DBST database is there. So many databases are there. These are connected with the BLAST. And for the protein, that PRF, PDB, SwissProt, GenBank, also many other are there. So it is connected with that. So when you are doing the blast, I mentioned we can go directly from the NCBI. So if you are opening the homepage of the NCBI, immediately you can see the right hand corner first option will be the blast. You can click on this blast. Once you are clicking, it will be taking to you this particular page. So T blast 10, T blast X, etc. is there. And if you are directly typing in your Google that blast, and the different way that homepage will open. But anyhow, you can able to do your experiment in both the cases. Fine. So I have projected here that how with respect to that particular Google, okay, with respect to the particular NCBI positions, how you can open the blast. So you have opened the blast. So this is the activity. Suppose with you are having the protein. So you are clicking on the blast P. You just look at here, blast P is mentioned. You just click on the blast P. So the blast P page will be open, and you can paste your protein amino acid sequence over here. So after putting the amino acid sequences here, so here you can direct. Somebody's microphone is on. Kindly switch off. Somebody's microphone is on. Kindly switch off. So you are putting the protein sequences here. That is your query sequence you are putting there. Then from here you can select the database, which database you are looking for. Otherwise, just leave it by default. And here, what type of blast algorithm you are going to use. So that also you can select. Otherwise, that also you can leave it by default. And just click over in this blast. Once you are clicking, you are going to get, this is respect to the protein, this kind of page will appear. It is going to indicate when you are providing the protein, the domain wise it is going to compare, because protein's domain is the functional part. All the protein try to conserve amino acid in the domain part. That's why domain to domain comparison is feasible for the protein sequence. So you are going to get this kind of result. And further, if you're going down, this is called the graphical presentation of your result. And this is the having certain kind of score greater than 200, 80, 200, etc. This is the graphical presentation we used to tell. And if you click on all this particular bar, immediately it will be taking you to the corresponding alignment positions or the alignment page. Fine. So the length of this bar, it is indicating that finally how much similarity is there between the database sequence and your sequence. And this bar, the shortness of the bar, it will be indicating the partially matching, and the total length of the bar, it is going to indicate how better way your sequence and the database sequence is going to match. And based on this matching, that particular color will appear. And the color is going to indicate this is the similarity pattern or similarity position, similarity score. We cannot scale, it is actual score. This is a kind of scale, okay, color scale. And if you click on this particular, any of this line, you are going to get, this is the particular alignment. So in this alignment, the plus is indicating similar group of amino acids are there, and DD, if it is matching, here is D, here is D also, it is matching. So like that, whether it is a similar amino acid or where is matching, where is not matching, all this aspect will be provided here. And finally, with respect to this alignment, it is going to calculate the bit score, expected score, and then how many alphabets got matched, that percentage, how many gap provided, that percentage. With all this aspect, they are going to calculate with the specific BLAST algorithm. And this is the important things we required to understand whether my protein, it is having what kind of similarity with the databases so what with other kind of protein. So statistically significance values we can see here. Here 40 minus 80, these are all statistical parameter. So we need to understand what is the statistics actually telling. So here we are getting the e value. E value stands for expectation value. Okay, so expectation value, it is indicating the probability that the result in alignment from a database search are caused by the random chance. So probabilically random chance, it is the random chance to match with my sequence that particular position alphabet with the database position alphabet. It is going to compare with the statistical significance value. And basically the e value, it is depending on the p value, that is the normal distribution curve, whatever is there in the statistical field. It is indicating that particular normal distribution pattern of the p value itself. So E we can calculate by multiplying with M, N and P. So what is the M? M stands for total number of residue in the database. N stands for residues present in the query sequence, and P stands for the probability pattern or the probability score. Okay, that is called high scoring segmented pair. So I mentioned high scoring segmented pair, whatever you are getting maximum. So that value also need to be calculated here, and that's where we can get the E value. So for example, if you are having a query sequence is having 100 residues and a database having total 10 to the power 12 number of residues, and with that respect p value, if it is like 1, so the resulting p value for the ungapped HSP region of 1, if it is there, then the database matching value should be 1 into 10 to the power minus 20. So these three value, if you are putting together, we can get the E value. So E is equal to 100 into 10 to the power 12 into 10 to the power minus 20. So all this, if you are doing multiplying together, it will be coming 10 to the power minus 6. And this 10 to the power minus 6 will be written as a 1E minus 6. Okay, 1E expected, 1 expected and minus 6. So 1E minus 6. So like that, you got the value here 4E minus 80. That means definitely the value was to the power 80. Okay, 10 to the power minus 80. Okay, 10 to the power minus 80, it was there, and the 1E, it is indicating this match, so it got multiplied by the 4, and that way the value is coming 4E minus 80. So this is the completely one statistical data, I must say. But here is having one problem. The problem is it is going to every day basis, every hour basis, the databases of what are the nucleotide sequence or protein sequence every researcher are depositing, the day-to-day basis, hour-to-hour basis, it is going to increase. That means now with the one particular sequence, if you are calculating the E value, after 1 hour or after 2 hours, if you are same database, if you are using same protocol, if you are following, you can may have or you can may get the different E value. So that means this E value can be changed. So as the E value is going to change, so slowly, again, one more pattern we have adopted. So before coming to this pattern, I just want to say that what is the significance of this E value. If E value is coming 1E minus 50, E is lower than 1E minus 50, so that is indicating these two sequences are having homologous relations. If E value is coming 0.01, that means 1E minus 50, if it is coming 0.01, then the result will be the partially homologous these two sequence. And the E value, if it is falling between 0.01 to 10, that is, we are going to tell there's a no significant similarity between these two sequence. That means they are not homologous also, and they are very much with respect to the distance tree, definitely they will be very apart from each other. And if we get the E value greater than 10, we can tell either it is unrelated or by chance that is having a similarity or relationship between these two sequence. So E value, we need to keep in our eyes on the E value. We need to understand the E value, and then only we can able to tell this which alignment is the better alignment and which is useful for your research purpose. And as I was telling, the E score is having E score is depending on the number of sequences or alphabet present in the database. So that is going to give kind of changes value keep on. Okay, so to overcoming that problem, we have considered the bit score. So bit score is completely independent of the your database, how many number of alphabet you have kept on that. So bit score completely generating with the help of lambda into s minus log of k divided by log of to the base 2. So whereas lambda is indicating the Gumbel distribution. Just a few minutes back, I was telling the probability distribution, whatever you are accepting here, in the mathematical, that particular model is called the Gumbel distribution. In the Gumbel distribution, there is having a specific value. If it is the E value is falling between this and D, in that case, the Gumbel distribution score should be constant. Actually, Gumbel distribution, it is relying on the P distribution. Okay, probabilistically distribution, that standard graph, whatever is there. With respect to the standard graph, each and every coordinate of the graph will be having a specific standard value, and that value is called the Gumbel value. So once we are aligning the these two sequence, whatever the information we are getting with the Gumbel distribution value, we are comparing it, and that way we are getting the bit score. And if we look at here, there is having a particular bit score. So it is written score 257 bits out of 657. So this bit score to be constant, because bit score is never ever going to change, and bit score is not depending on the total number of sequence present in the database. That's why bit score is very, we are also considering the bit score apart from the E score. So output format, as you have seen, the red, green, blue, different color annotation was there. So red part is indicating most related sequence and most related matches they found. Green and blue is indicating moderate to related. So green is having maximum weightage than the blue. So green and blue both are indicating the moderately related, whereas the black part is indicating, if the line is coming in the black line, okay, the ink should be present in the black, if in the line. So that is indicating this particular part of the sequence is completely unrelated. Fine. And that way we can get this kind of graphical presentation. And here one more thing we need to understand. So once we are clicking on the first, when the result is coming, we are getting this kind of one after another with the sequence, sometimes accession number, sometimes some reference with that, and then we can get the E score and some kind of hits. Hits is indicating total number of residue to residues, how much it is covered. That is called the hit score. And with the respect to that, E value just now I mentioned, they will be having the E score. And below that, they will be having in this alignment, when you are clicking, when you are seeing, looking the alignment. So first will be there, will be having a header part. Header part, it is which particular sequence, with whom it has been compared, etc. It is mentioned. And then statistical score, it is provided, bit score, E score, then percentage of identity, positive value, negative value, etc. And then the alignment part will be there. So this is the output basically we get in the FASTA and BLAST, and that's why the BLAST is very, very much useful. And you need to understand what is the bit score and what is the Z score. And now another 5 minutes I'm just taking just to finishing the FASTA part. So FASTA, it is called first of all or first all, and this is the URL for it. As I mentioned, FASTA created first, and then as the FASTA taking a lot of time, so BLAST, that approach has come. So FASTA here, similar type of algorithm FASTA also using. In the BLAST I mentioned for your protein 3 alphabet, and for nucleotide in 11 alphabet we are considering to making the word. And here in the FASTA, it is the number of alphabet they are considering very small. So in case of the FASTA for protein, it will be the 3, no problem. But for DNA, it is most probably the 6 or 5 to 6 residues generally they are considering. So if you are considering the 5 to 6 residues, definitely it is going to take much time. And if you comparing with the 11 residues, so that's why there in the BLAST it is called the word to word comparison. Here it is called the K-tuple or K-tup comparison. Okay, K-tuples or K-tup comparisons. So what exactly FASTA is working? How exactly FASTA is working? Suppose this is your sequence and this is the another sequence. Suppose sequence 2 is your query sequence and sequence 1 is the database sequence. So when these two are going to compare, so first software what it is doing, they are looking, they are first placing all the alphabet. Okay, this kind of table first will be created. So in the table, first they are going to write all alphabet, whatever is present in these two particular sequence. Now, if A, okay, this first alphabet, suppose A, in the sequence 1, how many positions or which particular position the sequence A is present. So here in the first sequence, the number 1 position A is present. Whereas if we look at the second sequence, 1, 2, 3, 4, 5, 6, 6th position, this A is present. So like that D. If you consider the D, which particular position D is present? 1, 2, 3, 4, 5. Fifth position D is present, whereas sequence 2, D is present fourth positions. So like that, each alphabet they are going to find out with respect to these two sequence, and then they are going to get the offset value. What is the offset value? They are going to look at the sequence 2 minus sequence 1. Okay, that value whatever is getting, that is called the offset value. If you look at here, actually 1 minus 6 they are doing, so minus 5, 5 minus 4 1, 6 minus 1 5. So like that they are calculating the offset value. When and after getting the offset value, need to find out that which one is the maximum number. Okay, which particular line wise it is having maximum offset value. So here with respect to the D, offset value is 1. With respect to the P and S, offset value again 1 and 1. So you need to consider this 1 and 1, this particular value, and then you can able to arrange this particular sequence. So here 1 is with respect to the which particular two alphabet. So S and if you look at the this particular number, offset number is important, and apart from the offset number, these two position also need to look at. Okay, third position and the second position, what is there. So that way, if you are putting this particular sequence, so PP is matching, then SS is matching, DD is matching, and rest of the part is not matching. So this way, the first it is continuing the calculation with the help of algorithm, and finally this kind of sequence informations it will be appear in front of your monitor. And here also that dot matrix as well as the dynamic matrix, both are going to useful, and both are going to use background the particular software, and finally we are going to get this kind of result. So in the first here we are getting the in the e-value with respect to the beat score, whatever you look for the blast, here we are going to get the jet score, and the jet score, if it is coming greater than 15, then it is a extremely significant correlation. That means homologous relation between these two sequences are present. If the jet score ranges between 5 to 15, then is having highly probability for the homologous these two sequence. Whereas jet score, if is lower than 5, it is telling this is the less certain. That means there is no relation between these two sequence. And for doing the first term, you need to use the European Bioinformatics Institute, this particular website, and you can type on the Google FASTA. This particular page will appear. You just click on this FASTA, this particular first hyperlink positions. So if you're clicking on it, you are going to get this is that home page, and here so again which particular database sequence you are looking for matching, so you can select from here. Otherwise here also will be having one option, which either protein or nucleotide, which sequence you are going to paste. So if you are taking the protein, so directly need to paste your protein in the FASTA sequence. So you are going to provide the protein in the FASTA sequence, and then you need to provide your mail ID and just clicking off. And once you are clicking in your mail, the result will appear. If you are clicking on the hyperlink, you are going to get these are the sequence with whom it has compared, and lengthwise how much it has covered, what is the expected your score. Okay, statistically what is the score, everything it will be mentioning over here. And you just click any one of these particular table related informations, you might able to get a lot of sequence related comparison part. So this is the sequence related comparison part. And again, if you're clicking on the output, so you are going to get this is the kind of result. So anyhow, only this much important for us to understand what kind of alignment actually there, where exactly the matching parties is there, all those. So this is all about the FASTA, and I'm repeating, it is very important. Once upon a time, it was having a very good important. Now, none of us is doing the FASTA. Always we are looking for the blast. And the FASTA also is having, this is the color annotation, your graphics card mode representation, it is called. So here also this line is indicating that how much similarity is there between these two score. And red, like the I already explained in the blast what the color is having significant there, red, blue, green. Similar significance also it is present for the blast. So red is indicating 100% matching, then orange it is almost like 70 to 80%, then green moderately matching. And if you look at there, this particular side is having lot of gap, whereas this up to this much position there is a no gap. So you need to understand that which one is your perfect alignment giving. So for knowing that, you need to know what is the Z score pattern or Z score value here. By looking the Z score, you will be confirmed. So this is all about your sequence alignment, and normally sequence alignment how is to do along with the first and blast if you are doing. So that is also doing the pairwise alignment, but what type of algorithm they are using and what purpose we can use the blast and firster, I have explained everything. So if you are having any queries, please ask me question. Thank you.
I have one question.
Yeah, please.
Yeah. That is when we should select pairwise as well as when we should select global alignment. Sorry, when we should select local and when we should select global alignment. Is there any criteria to select?
No. There is no criteria. If we wanted to, especially if you homology modeling purposes of the protein, there only particular segment, particular domain, exactly if you wanted to compare, that time we basically look for the local alignment. Otherwise, always we prefer to go for the global alignment only. Okay. So only with respect to the protein, it is better for the homology modeling purposes, go for the local alignment. Otherwise, for nucleotide, there is no actual, of course, choice. I hope you got the idea, sir. Both the method, any method you can do. Only thing you need to specify, this is the particular algorithm and this is the particular process I have followed. Nothing else. Nothing else.
So I have a doubt.
Please ask me.
So in the starting of the lecture, you have showed us a series of sequences in which it represents conservative, semi-conservative, and non-conservative. Can you please explain how we can classify it, sir? Okay.
Conservative, semi-conservative, I was talking with respect to the protein. So these are the amino acid might have, they can replace to the each other. Or these are the amino acid, basic amino acid, they can replace to each other. Because in the nature have selected them already. Fine. Now, if similar kind of protein, if you are arranging, okay, these are the, suppose histone protein, okay, histone 1. If you are arranging one after another, and there you try to find out whether position wise in same protein, but in the different organism it is present, whether position wise the alphabets are same in all the organism of that particular protein or not. That is the meaning of conservative and semi-conservative. Here conservative means you can see here only SS and AA. That means S and A, if you look at the criteria of classification of amino acid, they belong to the same group. That means their property, okay, physicochemical properties are same. That means though it is the histone protein and it is found in the different organism, but the amino acid replacement will be the from that particular group only. In that way, we can tell this is the conservative. Non-conservative, if we look at here, LLPPP, it is there. So human and chimpanzee, higher organism, same protein, having different group of amino acid. Whereas mouse, rat, and cow, they are having similar group of amino acid, but L and P, of course, not belonging to the same physicochemical property wise in the same group. That means this is the particular length wise, this is the particular line wise. If we look at the amino acid sequence, we are getting lot of difference. That is the meaning of non-conservative. Fine.
Yes, sir.
Semi-conservative, it is again with respect to, I mentioned for non-conservative, completely two different group of amino acids. Okay, and semi-conservative again, suppose here total 10, if we consider here 1, 2, 3, 4, 5, 6 alphabets are there in this particular line. Fine. Among these 6, suppose we found the 2 belonging to the one group, same group, and VV belonging to the another same group, whereas another amino acid is belonging to the third group. In that case, we can tell this is the semi-conservative. Means few of it is conserved and rest of it may not conserve. That is the concept of semi-conservative.
Okay, sir. Thank you, sir.
Welcome.
Sir, there is a question from Gurjant Singh. He has
Yeah.
posted in the chat box, sir.
Yes, yes.
Sir, the adenine, thymine, and guanine cytosine ratios are close to 1, whereas the adenine guanine ratio varies considerably. Why is it so?
Sorry, I'm not getting it. It is there in the chat box.
Yes, sir. It is written in the chat
I
box.
just got to it. Sir, the adenine, thymine, and guanine cytosine ratio are close to 1, whereas the adenine guanine ratio varies considerably. Adenine guanine ratio varies considerably. I'm not sure, but adenine, as I think it is as per the Chargaff rule, something it is there that ratio should be 1. But how it is, why it is deviating from the Chargaff rule, I'm not sure. Adenine guanine. One reason it could be because the higher organism always try to show the complexity in their genome. So in that case, G content should be more than the A content. And G and C, G if it is there in the one stand, definitely other stand will be having C, and between them will be having 3 hydrogen bond. So eukaryotic organisms are having more complexity, more repetition, more G content, and then any prokaryotic organism. Might be that could be one of the reason. Might be that could be one of the reason, because G and C always it is exhibiting with the COT curve value, TM value exhibition calculations, and genome complexity, that calculation also. So higher organism definitely having more G and C than the A and T. I think this is the answer.
Yes, Swarnalika, if you have any doubts, you can unmute and ask your doubts.
Yes, sir. Thank you, sir. Am I audible, sir?
Yes, yes, you're audible.
Sir, today we are talked more about the protein-protein interaction, sir. I'm having some doubts in that. Basically, we are while we are comparing.
1 minute, 1 minute. I am not mentioned anywhere protein-protein interaction. That is completely different aspect. I mentioned only protein-protein comparison. Interaction and
Sir.
comparison completely two different meaning. Yes, you continue.
Yes, sir. My first doubt is you're at the starting of a lecture, you gave that there are 20 amino acids and each is started with the capital letter. Why only one letter or only capital letter? Is there any specialty among that?
No, it is completely basic biochemistry. Biochemist peoples, they have mentioned that amino acid can be exhibited by this way. And if you're writing the 3 alphabet, might be software, it will be taking a lot of space. Fine. So every software that is having one particular word for all aspect, not only the software, anything, if you're writing anything in your word file. So in one particular sentence, you can maximum 20 or 30 alphabet you can put, not more than that. Fine. So that space is very limited. We can tell with respect to the software that is the K teaming value. Okay, that means cut shorting your space. So last class I was telling that gene sequence. Okay, I have shown that 60 alphabets are there, because the software made in such a way that only one line will be having 60 alphabet, not more than that. But that means the space is very limited. So that way, for all kind of software in the bioinformatics field, whatever you use, there always we prefer to put the single capital alphabet for the protein. I hope
Yes.
you got the idea.
Yes, sir. My second doubt is why the similarity in homology and gaps are to be the important.
Why?
We talk about similarity, homology, sir. Why they are playing an important role?
Yeah, gap is playing important role because I explained the dynamic matrix, right? In the dynamic matrix, when you are giving or when you are comparing the sequences, so automatically the gaps will be created. Automatically the gap concept will come. Got it? Automatically it will come. So though initially I mentioned the gap, we need to extrapolate the gap, but why this extrapolations or everything is there, that is completely mathematically proved in this dynamic matrix.
Yes, sir. My next doubt is, as if we are going to compare any two organisms, or we can compare anything, mostly DNA and RNA were used, what I learned before. But we are talking about protein comparison. Is protein comparison is important such that of DNA and or RNA?
Yeah, protein, see, protein is the outcome of the any gene, right? Any
Yes,
gene,
sir.
that is the DNA, if it is there, then it is translating to the mRNA, from mRNA to protein will come, right? Now DNA total composition, definitely it is not going to translate into the protein, because for eukaryotic system a lot of intervening sequences will be there, that will be removed, then functional mRNA will be created, then it will come to the protein. And protein, it is basically mediating the function of the gene. We used to talk the gene, this gene is present, this gene is responsible for that. That means the gene is encoding some protein, protein is mediating that function, right?
Yes, sir.
So if we wanted to know directly to the molecular level, there is a two different aspect is there. Either you need to find out the protein, what kind of amino acid changes are happening, so that this defaulting activity the protein is playing, right? And when we are able to trace that amino acid, you know that amino acid could be encoded by so and so biological codon. So the codon exhibiting the mRNA, and that again exhibiting the DNA level. Right? So nowadays, for every purposes, rather than DNA, we can go for the protein. And the protein modeling, all those aspects, definitely look for the protein sequence. So protein, if we are looking, all the information related to the DNA we are going to get. Understood? But if we exactly looking to the gene level, that is the DNA level, so the DNA, you can predict this maybe this kind of protein it is going to encode, because you are telling maybe, right? So some differences might have come in the original biological level. So that is the difference. Always we look for the protein-protein comparison to better understand about the gene function.
Yes, sir. Thus, the mutation in the protein, you can say that proteins are also having the mutation, plays an important role in any evolution or any that parts like that.
Yeah, protein, of course, actually, I'll be talking this particular concept on the phylogenetical tree. There, we give the emphasis, we are selecting such kind of protein, the protein which never ever go for any kind of mutations. So those are the marker protein. If you're taking those marker protein, we can construct the phylogenetical tree, we can understand the relation. But if you're taking some protein which are very abruptly is going for mutational aspect, so that time it will be difficult to predict anything. Okay, so you cannot able to correlate with all the things together. And protein, if the amino acid is going to mutate, then the effect definitely means in that aspect. One point you must know, that is called Obel hypothesis. All the point mutation for the protein is not deleterious, right? Because in the biological codon, the first two alphabet is important and third alphabet can change. That's why tyrosine and other amino acids are having 2 or 3 different codons. So all the points mutation is not important. What is important? That point mutation, it is really deleterious for the protein or not. That we are going to find out with respect to the protein. I hope you got the answer.
Yes, sir. Thank you, sir.
Yeah, welcome.
We will start off with the BLAST and FASTA search. So in the morning, so it was in detail, the professor explained about the BLAST, the types of the BLAST, and then why we should use the BLAST or the FASTA. So we will do with the hands-on now. how we can perform the blast search, and then how we can do the faster search, how we can interpret the blast results, and some of the advantages and the disadvantages that are there in the blast, and also the faster. And we will also look the parameters. There are different parameters that are there within the blast when we perform. How are they affecting the alignment? Affecting in the sense which parameter we should change to get a desired result. So that's what the affecting term is referring to. So again, we will go slowly, and we will look for understanding the purpose of this blast, though it is done in the morning, but so we'll go and exercise this for better understanding. Now you can just type blast in the Google, and then we will have it like basic local alignment search tool, and then click on this one, basic local alignment search tool. Now, as we already performed some experiments yesterday, so I will just ask some questions again, and maybe if you start answering for that, okay, it becomes very easy. And also in my previous class, I have asked few questions, right? So you can also answer for those questions at the appropriate time. Now, as in the morning, right, as discussed the different types of the blast programs. We have like blast 10, blast X, T+10, blast P. Okay, so these are the different blast programs that are there. Now the purpose of having these different blast programs is also discussed. So the purpose is, if I have a nucleotide sequence, in other words a DNA sequence, and I want to see the DNA sequence matching to my DNA sequence, then we go for this nucleotide blast. If I have a nucleotide sequence, but I want to see to which particular protein it belongs to, then we go for this blast X. If I have a protein sequence and I want to see to which particular protein it belongs to, then we go for this protein blast. And if I have a protein sequence and I want to see to which DNA sequence that it is matching, then we use this T blast N. Another version is also referred as T blast X. Okay, now we will do it one after the other. Now, can you tell me, if I want to do, we will start with the protein first, okay, then we will go for the DNA. Now, can you tell me, if I want to perform the protein blast, and if I click this one, for example, protein blast, I want a sequence. As we know, we are working with the human CCR5. So as I mentioned, we will continue to work with the same protein so that we will better understand that particular protein. And as I mentioned, different platforms like BLAST, FASTA, pairwise, multiple sequence alignment, phylogenetic analysis, we will appreciate what kind of a data information we are going to get. Now, from which database I will get the protein sequence? Maybe someone can unmute.
PDB.
PDB.
Okay, I will repeat the question. Okay, from which database we will get the protein sequence?
Uniprot.
Correct. So we will get the protein sequence from the Uniprot SwissProt. Now, even in the last class, I asked a question like, what is the difference between the Uniprot SwissProt and Uniprot Tremble, TREMBL, right? So maybe anyone can tell me what is the difference?
TREMBL is the translated nucleotide sequences which are being deposited in the Uniprot. So that's the TREMBL.
Correct. And then SwissProt is?
protein sequence directly from the sequences.
Curated sequences, these are.
Curated sequences, perfect. Tremble are not curated, are unreviewed. And if you look into that one, so it was mentioned like Swiss prot is a reviewed and a Tremble is a unreviewed, right? Now that means the Swiss prot refers to put it into a simple terms, the chairperson has looked into the 5 faculty members reports and then made it one page write-up and then given the data. That process is the review. So that data goes into Swiss prot. But there are 5 members who has written the write-up about the department, but the chairperson has not yet made the 1-page write-up. Okay, then those 5-page write-ups, where should it go? Right? So that will go into Tremble. I hope it is clear, right? So there are 5 members. Let us say the Department of Botany as example. Now in the new college, 5 members, they have written the write-up and the chairperson or the HOD collected, reviewed, and then made it into a single page report. And then that data, whatever submitted by the chairperson, that goes and stores into Swiss prot. But in other college, okay, again the Department of Botany, for example, there are 5 faculty members or 10 faculty members. They have also written the write-up, but the chairperson or the HOD has not yet made the review of these 10 members report. But just because the chairperson or HOD not reviewed, can we say like the data is not available for that particular department? No. So what they did is let the faculty members deposit whatever the information they have into one database, and that database is referred as Tremble. Once the chairperson looks into these 10 reports and then collects, makes it into a 1-page write-up, then that goes into back to Swiss prot. Is it clear? The difference?
Yes, sir.
Yeah. Now.
Yes, sir.
Very good. Now, once the chairperson or the HOD makes the 1-page write-up, whether the file will still be there in the Tremble or not.
Until or unless it is submitted, it remains in the Tremble.
It's not audible, sir.
Hello?
Yeah, bit louder, sir.
Yeah. Until or unless it
Yeah.
is submitted,
Yes.
it will not go to Swiss prot.
Yes. Now, once the data is submitted by the chairperson to the Swiss prot, right? The review, if it is done, and once it goes into Swiss prot, the Tremble data will be removed. Okay, because it becomes duplicate. But if it is not there in the Swiss prot, the data will be there in the Tremble. Okay. So that is how the hierarchy is maintained. Because once the HOD is already made a note of all 5 reports from the faculty members, again retaining the data in the Tremble becomes redundant, because already we know millions of data is already there in the Tremble. So then they will make it like remove from the Tremble, because the chairperson will cite first one, second one, third one, fourth one from these faculty members and then send a report to the Swiss prot. So then there is no need, because already the citation is done. That's how the process is. Now, when we want to do the blast P, okay, so we need the sequence. So you can easily see enter query sequence. Since it is a blast P, we got to give the protein sequence. Now we also know the protein sequence we get from the UniProt. So go to UniProt. Then you can type either human CCR5 or P51681 is the accession number. Okay, so this is what I am mentioning. The Swiss prot we have around 5.5 lakhs, But Tremble, if you look, we have around 21 million or 2.1 billion entries are there. So once it goes into SwissProt, the entry will be removed from the Tremble. That means so many faculty members have submitted their report, but yet the committee has to review and then convert it into or submit into SwissProt. That's what it is giving. Now here you type human CCR5 and then P51681 is the entry. You click onto this one. Then we want, for any experiment we do, okay, just look into the format. What do they accept? So here they have clearly mentioned either we can give the accession number or gene IDs or FASTA sequence. So my recommendation is always give the sequence, because the accession number may go into a different versions. Usually it will not change. Accession number, once it is assigned, will not change. We will have only the newer versions available. But to avoid all the confusions, it is always safe to take the FASTA sequence, because we are very sure that yes, it is human CCR5, the ID is there, and we can also see that it has 352 amino acids. If I go to the sequence, is 352 amino acids. Okay. In the previous class, I asked one more question, that in the NCBI, from 13 to some number, some base pairs were there, and the length in the amino acid, it was 352. So is the number of nucleotides and the number of amino acids are matching? Any chance anyone worked out on that one?
No, sir. Introns are there.
No. We will not have introns, because when we looked into NCBI and we worked out with the human CCR5, by any chance you can tell me the accession number?
P51681.
No, that is the UniProt ID. Okay, so that's where this is P51681 is the UniProt accession number that codes for this particular amino acid sequence. But the DNA sequence we will get from the GenBank. And in the GenBank, we took this particular entry, complete code yes, that is the coding region. Then we clicked on this one. Then we worked out saying like from ATG, this is where the coding region is. Once the coding region starts, so we will not have any introns. It's a complete coding region, one exon.
Yes, sir. The result is you have got from 13 to 1071.
Yes.
By deducing from 1071, it is 1058, and then it is divided by 3. The number of amino acids is 352.66. That is near about 353.
Yes.
So our result is more or less same. 350 you have said.
Yes. Now, if you count this way, because we have to add 1. So if we count from here till end, we will have around, I will just tell you, when you we will have around, we will have exactly 1059 base pairs. From here till here, we will have 1059 base pairs. Okay, now 1059 base pairs corresponds to 1059 divided by 3. That corresponds to 353. But we do not have 353, but we have 352 because
yes, sir. Last one is that termination codon.
So this TGA is actually the termination codon. So for the termination codon, we do not have any code to represent in the sequence. Okay, usually it will be given like a star, but in the sequence we will not have anything. So it just says like end 352 it becomes. Now why I made you to do this small exercise is, whenever you do the sequencing and get the sequence length, and if somebody says like coding region, then you should know that the last 3 letters codes for a stop codon, and the stop codons are UAA, UGA, UAA, UGA,
UAG.
AG.
UAG. So we have these 3 stop codons. So just check whether we have these 3 stop codons in the given coding region. If it is there, then subtract that last codon and then take a count, and then tell like my gene is having so many amino acids. So the calculation becomes very clear. And we will not have anything like 352.6. Either it will be 352 or 353. We just need to add +1 because it starts from 13. So 1, 2, 3 like this. So if we just do the minus from this to this, again +1 we need to do. So we will have the exact count available. Now take this FASTA sequence. Okay, I will just zoom in. Copy this sequence and then go to this BLAST, then paste it over here. Now we already know the significance of this FASTA format. It starts with a greater than symbol, then we will have a 1-line description. So the advantage of this is, because the tool knows that this is a description about the, so we will have the job title automatically fetched. We don't need to type here. If it is not there in the FASTA format, for example, if I remove, okay, if I just submit the plain sequence, okay, I have just reset. If I just give the plain sequence without that greater than symbol, so job title will not take it by itself. We need to manually type something. Okay, so to avoid that, job title, this is something like job description, okay, what it is. So take the FASTA sequence and give it over here, then automatically the job title will be taken care. Now we have another option here like query subrange. Any chance someone can guess what it is, query subrange?
Yeah, from which sequence to which sequence we can limit, from suppose 50 to
Exactly.
850.
Perfect.
Like that.
Exactly. Now I know the sequence is having 352 amino acids, but if I don't want to cover some portion, this is the 352. But if I know about the, because the description is given in the UniProt and functionality is given, we also have the structural information and where exactly the helix is there, where exactly the motif is there. So all those description you can easily make out. So this is the extracellular, then the first helix, then the cytoplasmic, right? So if I have to draw this structure, right, you can also do this. Maybe I will just open a paint. Why the quick lines here? Okay, let me place it here. Why, when that particular option becomes useful? So if I have to convert this sequence information into 2-dimensional structural information, we can easily make. Let us start from here. Maybe the thickness we can increase. It's visible, right? So you start from this, then go like this, like this, like this, this, and like this. Now this is 1, 2, 3, 4, 5, 6, and 7. Now what I am doing is, we have a extracellular, right? We know that we have a membrane protein. This is the extracellular and this is the intracellular, because it is already given where it is present. So this is on the cell membrane protein on this. That means the protein, some portion is intracellular, some portion is extracellular. Now whether it is matching with the sequence, that's what I'm just, you can also write down in the book. Okay, this is one of the interesting ways by which you can understand. This is extracellular and this is intracellular. Now this shows this is extracellular. Whether is it extracellular? Yes, they have already mentioned like it is extracellular. And from where it starts, so the first position, first amino acid to the 30th amino acid is extracellular. Then from 31st amino acid to 58th amino acid, 58th amino acid is the helix. So that means this is our first helix. Then it is intracellular or cytoplasmic. Yes, the third one is cytoplasmic, starting from 59 and till 68. 68th position is the intracellular. Then second helix, yes, helical name 2, then extracellular, then helix, then cytoplasm, then the fourth helix, helical number 4. Then after this, extracellular, then fifth helix, helical, cytoplasmic, sixth helical, then extracellular, seventh is helical, and then the cytoplasmic. So this is the cytoplasmic, 302 to 352. So that means here 302 to 352. I hope you are able to understand, right? How is the correlation between the amino acids, the structure, and its description. Any doubts with this? Anyone?
No, sir.
Okay. So for any protein when you are working, right, especially the membrane proteins, so by understanding this, it becomes very easy to know which portion acts like an intracellular, which portion acts like an extracellular, and which region. So let us say if somebody says like the amino acid which is there from 130 to 140th position, where do they lie? So from 130 to 140 means that is this region, 130 to 140. So that is cytoplasmic. Then you can say like that goes into a cytoplasmic region. You can easily say like where it is, even if the description is not there. For your protein, you can make such structure 2D representation and then you can easily explain. Now coming back to the blast and these information, if I know that the first 30 residues and the last 50 residues here are outside, so the first, this is a completely loop region, and also this region I am not interested in. I am interested only with this from here till here. If that is the interest, that means from 31 till 301, then give that region 31 to 301. If you want to do a very specific search, then you will go give this query sub search. If that is not the criteria, keep it as it is. Okay, so at this point of time, keep this as it is. Now in the database also we have different types of database that are available. That means in which database it has to search. Choose search set. That means whether it has to search in the non-redundant protein sequence, because the query sequence is protein. So whether it has to search in the non-redundant NR, that means entire database. or you want to give anything with reference to RefSec select proteins? By any chance you can recall when we use this term RefSec, where it was earlier used? Anyone?
CBI, sir. Theoretical sequences are there.
Yes, in the NCBI, when we type like human CCR5, right? So this entry is from the RefSec, reference sequence, RefSec. Okay. So whether it has to search in such entries, not in these. Searching in all these entries is the non-redundant. Searching within this database is the RefSec. Whether it has to search in a reference or model organisms, SwissProt, or you want to search in patent or structural database, PDB. PDB is a structural database, okay, not the sequence database. And then metagenomic or transcriptome with assemblies. So we will use the SwissProt now, okay, because we want to see, by any chance, anyone guess what is the use of performing this BLAST P? What should we do?
Protein to protein comparison.
Correct. Protein to protein comparison. So the purpose is to know whether the sequence of human CCR5, which is of this 352 amino acids, is there any other organism having the same exact 352 amino acids? Now, why should we find that one? If it is there in other organism, let us say if we find the same exact 352 amino acids in the mouse, and mouse being a model organism, so one can easily use that organism as the model organism to do some experiments. Now again, what kind of experiment? Because CCR5 is involved in HIV/AIDS and I want to develop a drug molecule. I want to make my drug molecule bind to this CCR5. Since I cannot use the human as a trial in the beginning, I have to use some organisms. So since the same protein is present in the mouse, so the chances of the way drug acts in the mouse remains the same in the human because the protein remains the same. So to know such results, where exactly it is matching, we will use this BLAST. Okay. The purpose of using BLAST may vary from the project to project. I will give all possible ways or possible reasons why we use this BLAST over a period of time. Tool. Now we have selected this SwissProt. Now we have an option here, though it's optional, organism. Now we can also type the organism in which we want to find this particular sequence to be present or not. If we don't give anything, it is going to search all the organisms. If we want to specifically search in particular organism, that also we can do. Let us say if we just type like Homo sapiens, okay, that means we know that CCR5 is already there in Homo sapiens. But if our purpose is to find, is there any other protein in the human which is similar to CCR5? Are you getting? I want to know, BLAST is just going to check the comparison. Comparison is of two ways, finding the similarity and also finding the dissimilarity. Okay, if you are comparing two individuals, you are not only comparing the similarity between them, you are also comparing the dissimilarity between them. Okay, so two departments you want to compare. That means you are not just comparing, you are also looking at the differences between them. Okay, so in that particular context, if I want to see is there any other protein which is similar to CCR5 like CXCR4, CCR3, CCR2, is there any other protein which is similar to human CCR5 if I want, and it has to be within the Homo sapiens, then you give this. if your plan is, I already know that human CCR5, in other words, human is the organism, CCR5 is the protein, and I don't want any result where the humans to be there, because I already know it is there in the human. I want other organisms except human. Then you give the human and then click this exclude. So if you say exclude, it will search all organisms except Homo sapiens. So just a single click here will make the completely different way of searching. If you don't give this, it is going to search only in the humans. If you tick this, it will search entire other organisms except Homo sapiens. Okay. So for time being, let us say we want to see other proteins. Okay, organism is the same now. Other proteins which are going to match with the CCR5 within human. So we can give this. Then let's keep other parameters as it is. We also have an option down below. Later we can discuss the differences, but this goes into too much of details. In the beginning, it is better to just do with the BLAST P. Okay, this is position specific iterative, pattern hit initiated, or domain enhanced lookup time, or data blast. These things not to use. Okay, not required in the first stage in any of your projects. BLAST P is more or less well developed algorithm and will fetch the related information accurately. So then keep like this, and in the beginning when we are working, keep all the parameters default. They don't make any changes. Now maximum target sequences, that means how many sequences it has to show in the output. So take 100, E value, that is the expected value, 0.05, keep it as it is. So once we have the results, I will just explain you what exactly this E value is. Already in the morning the professor has explained, and then the word size, and then the maximum matches. He also mentioned the matrix PAM and Blossom. Let's keep the matrix as Blossom 62. Okay, don't change this one. Then the gap costs, this also we will discuss now once we have the results. Gap cost 11 and 1 as it is. Existence is 11, extension is 1. Okay, so keep this one as it is. Then the matrix that it has to use, we'll keep this in this also, conditional compositional score matrix. Don't make any changes in the first run. Okay, then select this, say like show results in a new window, then click blast. Okay, now we obtained the results, and the first hit. Hope you also obtained the results. If by any chance if you are getting any error or anything, please let me know. It's clear, right? You all obtained the results.
Sir, my FASTA sequence is showing error.
So if the FASTA sequence, if it is giving error, maybe you have not copied the sequence entirely. You might have copied from this. So this greater than symbol may be missing.
So should we get the FASTA sequence from NCBI?
No, you can always get, even in the NCBI here, right? Which one I took is this one. But the thumb rule is, if you are working with a protein, take the sequence from UniProt. If you are working with the DNA, take the sequence from GenBank.
Okay, sir.
NCBI. So that is why it is advantageous to have both the databases. And also in the previous class, right? So one person asked when we have the accession numbers available in the UniProt, right? We can go from UniProt and then go to GenBank directly from this. And also mentioned like that we can do, but the problem is, okay, if I come down, so if we want from the GenBank, we can just give this one, so the links get updated. But we will not have any information here, right? What exactly this is coding for, and we cannot keep checking each entry by opening it in a new window. So that becomes tedious. We have all the just the initial and then the converted mRNA translation sequence information, and we do not have anything like what exactly this entry is really coding for. So rather than checking like this, as long as we are working with the normal sequence, okay, not the mutated, as long as we are working with the normal sequence, so we can just go with this. Okay, even if I take EMBL, click here, and then if I open this, it will just takes me to EBI. So if I just update it to DDB GenBank and click the same link, this is going to take me to the NCBI. If I say DDBJ and click the same entry, then it will take me to DDBJ database. Okay, that's the only difference. And in the EMBL, that is how it is going to be showing. But the advantage, if we are working with GenBank, we will have the description given. So we will have more idea like what all the possible variants available or the work done related to protein. All that we will get from the GenBank. So in the first stage, type human CCR5 in the GenBank. Get to know what it is, what are the changes. If you have a RefSec, go to that RefSec and then take the information. You will have more idea about the type of the study that is done and everything, because understanding the protein and how much is done already related to that protein, that is extremely important. Then you go to the UniProt, okay, then take the next level, take the sequence of the protein and then start working. So that my purpose of telling everything is you need to be safe, okay. In other words, we should not make a mistake in taking the sequence, then it becomes a wrong thing. It's something like you already have a menu in the hotel, what you want to order, but still if you make a mistake in ordering, right, then that is not the problem of the databases. They are clearly mentioned everything, the menu, the cost, whatever, everything is mentioned. If it is just oral, then there is a chance of some having mistake. But when you have everything clearly specifically mentioned and just go with each of these databases, then you can make a decision what to use. Then this is how the result is, right? So now it is giving like in the score, we'll look into each of these things. First one, it is already giving like P51681 is the accession, and even we took the sequence from the same thing, P51681. So my query sequence belongs, because the BLAST does not know that I have taken the sequence from the UniProt itself. It just assumes that it is a fresh sequence. Okay, you have done a sequencing and you are submitting. That is how it is going to think of. Then it says like your query sequence is 100% matching, 100% identity with this particular entry, P51681, which we know is also human CCR5. Then which is the next protein which is matching to this? This is P51591. Now what is this protein belongs to? The description is given here. So this belongs to CCR2. Okay, if we want to go in detail, so you can just right click and this open it in a new tab. So this is CCR2 human. That means CCR5 and CCR2 human, CCR5 and CCR2, These are closely related protein. Now the length is, this is having 374 amino acids. What our CCR5 is having is 352 amino acids, right? This is having 352. This is having 374 amino acids. Now, this is where exactly the question from one of the participant in the morning asked, like, when should we use the global alignment and when should we use the local alignment, right? So that's the question now. Now, first question, whether the BLAST is a global alignment or a local alignment?
Local alignment as
Local alignment.
full name, basic local alignment.
Perfect, good. So many of the times people will forget the core of that one itself. That is there in the abbreviation of BLAST itself. That is basic local alignment. So many times they say like global alignment. The answer from your side would have been like global alignment, okay, if I have shown the output like this, okay. So 352 amino acids and 352 amino acids are exactly matching. Now this is the global alignment, right? Entire to entire is matching. But since I just showed you with this, right, so you are able to answer like local alignment. But it's correct understanding that it's a local alignment, okay. Now what it says is, when I performed with the BLAST, it says like the query sequence is matching with this CCR2. How much it is matching? The next question will be like how much it is matching. Then we will say like 79% identity, 87% positive, and there is 1% gap. We will also discuss each of these terms in detail now, okay. Identity refers to the exact match. Maybe I will just zoom in a bit. I hope it is clear now, visible. This one. Now, identity refers to the exact match. What exactly the exact match refers to is, in my query, there are two rows that are there. Okay, in fact, three rows. First row is the query. Query refers to whatever the sequence I have submitted. That is the human CCR5. So query is nothing but the human CCR5. Subject sequence refers to SPJ, that is the subject. Subject sequence refers to the sequence that the tool or the BLAST is giving. In other words, compared with. Subjects refer to the compared with. Now the comparison is made between two sequences. One is human CCR5 and another one is CCR2. Okay, that is this CCR2. And in the 13th position, okay, this 13 refers to, in the 13th position of my sequence, I have N. In the subject sequence, it is having D. Okay, why it is put plus, but why it is starting in the 25th position? Why not starting from the first position, right? Those are with reference to the BLAST algorithm, which professor has discussed in the morning. So just to put that into a simple terms, tyrosine and tyrosine is exactly matching, right? So if we look into this particular, always it goes with the column analysis, okay. Always we analyze on a column basis, and maybe if you want to, I can just take this into one PowerPoint, okay, so that I can mark and then explain. It becomes very easy. Last, let me take a share screen, copy this, and then I will put this here. Okay, then let me make this as a full screen. Okay, then I will use this. Okay, Now, whenever we do the analysis, please remember we always do on the column basis. This is row, okay, and this is column. Now, in the first position, what is there? In the 13th position, we have N, and we have aspartic acid here, 25th position. Then in the second position, it is matching. Y, tyrosine, and also the tyrosine, both are matching. Since it is matching and the nearby residue is positive, that is why it has considered. Then this region, when we do not have anything, this is referred as blank, not the gap. Okay, this is referred as blank. Blank means mismatch. Okay, blank refers to mismatch because the glutamic acid and alanine, they are not matching. The amino acids are having. What exactly that matching, or even in the morning, one of the participant asked, what is this whole story of this matching, mismatching, right? You say like conserved, semi-conserved, right? So you can just type like amino acids, physicochemical properties, okay, physicochemical. So all the matching or mismatching is made based on the physicochemical properties of the amino acids. Based on that, the classification is made. So if I had to show you, right, you can just look into this as one of the okay. Now if we look into this based on the physicochemical properties, what is that physicochemical property refers to? Isoleucine and leucine, they are the aliphatic amino acids. Phenylalanine, tyrosine, tryptophan, and histidine, right? They belongs to aromatic amino acids. All these amino acids, whatever is there in this red circle, they are all hydrophobic amino acids. Now that means if we have like valine, isoleucine, leucine, these three are aliphatic and also hydrophobic amino acids. So even I should be able to show you, there is one more a much better way of representing or explaining this, the properties, or I will open my slides. I hope this should be fine. We will understand more or less. Now this is the broader spectrum, hydrophobic, and these are specific. Now isoleucine to leucine, if they are changing, they are aliphatic and hydrophobic. But isoleucine to methionine, if there is a change, right, but they are not aliphatic, they are different. Let us say isoleucine and phenylalanine, for example. So isoleucine and phenylalanine both are hydrophobic, but they are not belongs to the same property of aliphatic. One is aliphatic, another one is aromatic. So for example, the values will differ based on how close they are. Just to give an example, if the isoleucine and leucine is there, then we say like both are aliphatic and hydrophobic, we will give the score as +5. But if it is isoleucine and phenylalanine, then we say like no, one is aliphatic, another one is aromatic, but they are hydrophobic. Let us not give the score as 5, but let us give the score as 3. Okay, because still the hydrophobicity is well maintained. Now let us say isoleucine and then the proline. Okay, isoleucine and P refers to the proline. Here it is. Now what value I should give? Then we will say like this is hydrophobic and aliphatic, but proline is the small amino acid, but that is completely different physicochemical property than isoleucine. So they are different amino acids because it is different. It is not there in this circle, but has gone into a different circle, not belongs to any of these things. Let's give like -5 for this. Then we say like they are different. Now, another example, if we look into D and E, aspartic acid and glutamic acid, both are acidic amino acids, right? And both are charged amino acids. Both are charged amino acids. So if aspartic acid to glutamic acid, if the change happens, we say like they are still one and the same. Okay, not much of a difference is going to happen if they get changed. So plus value we will give. That means the change is not going to affect a lot. So based on this properties, acidic, size, charge, polar or non-polar, sulfur containing like methionine and cysteine, these two amino acids are sulfur containing. Then the analysis is made. Then how the scores will be given for such stuffs is what this Blossom matrix has created. And in this Blossom matrix, they have clearly explained. It has not commented. Okay, there they made this analysis and then said like, let's give the score for any change that is going to happen. Now if we look into this aspartic acid and then the glutamic acid, we have a value like +2, because if we change glutamic acid to aspartic acid, not much of a change is going to happen. Why that change is not going to happen? Because both are charged amino acids. Now if we take this isoleucine and leucine, now isoleucine, right, is over here and the leucine is over here, right? Leucine is here and isoleucine is here, +2. Why +2? Because both are one and the same. Isoleucine, aliphatic hydrophobic amino acids. Now how about isoleucine to valine? Now if we look into isoleucine and then the valine, we have like +3 because hydrophobic amino acid. So looking into these physicochemical properties and the mutation rate and its effect on other amino acids, the values have been distributed. I hope it is clear, right? If the same amino acid change, it's something like there are two faculty members who is taking the same class related to botany, for example, right? So the effect of the teaching, how much it is going to vary. That's what these values are referring. So one faculty of the botany is not able to take, so another botany person will come and take the class. So how much is going to have the effect on that particular course? Not much. Maybe the same faculty, if he takes, let's give 8, okay, or let's give like a 8. But another faculty, but with less experience, if he takes, so let's give the value like positive value only, because he still knows the domain knowledge. So let's give the same value like 4 or anything, okay. But if there is another guy who is from the zoology background and asking him to take a class, how effective is that class? Then the class becomes a poor class. So how will you tell that it is a poor change? Right? It is a change. Whatever we are doing, one amino acid to another amino acid is a change or a mutation. So you made a mutation of the faculty from botany to zoology. Suddenly the class becomes poor. So you will ask the rating, how much I should give the rating. Then you will say like -3 or -4, because not able to, that is not the change acceptable. Okay. That change is not acceptable in the nature. So you will give the negative penalty to that. But change from one to another one, okay, let me come back to this, or I will open this PPT. If the value is positive, now to put it into simple terms, if we have the symbol like plus, that means that change is acceptable in the nature. So those plus will go into positives. Wherever we have like Q to H mismatch, then they will say like they belongs to two different functional groups or two different physicochemical properties. We cannot say like they are similar. Then you call them as a mismatch. That is how the classification is made. Then if it is identical, put the same amino acid here. Then one more question that was asked is why we have like a single letter, why not in the 3 letters, right? Imagine for the 352 amino acids, right? one letter itself is becoming so lengthy to interpret. So what if it becomes like 3 letters? Then it becomes too huge. So the representation here itself becomes too complicated. So to avoid that complication, and we already have this standard lookup table. So why you want to use like ALA or ARG or ASN, right? So that becomes more complicated in interpretation. So make it like a single letter. That is the convention why we have like a single letter representation, which makes our life easy to interpret. Now, we'll come back to this. Now, wherever we have these referred as mismatches, now we have around 4 gaps, right? These are the gaps. Now, any guess from anyone why we need to have these gaps?
Because in subject, there is no, in subject,
Yeah.
there is no sequence or no amino acid that is matching with the query. So gap is there.
Perfect. So the gap is introduced to optimize the alignment. Okay, the purpose of introducing the gaps is to optimize your alignment. Now, if I do not put these gaps, okay, let us say if these gaps are not introduced, then this WNN will come in this particular position. Now, if this WNN comes into this position, then from here onwards, nowhere, no residue is going to match. Okay, nowhere, no residue is going to match. Now, there are 2 purposes for which we introduce the gaps. How to interpret these gaps? It may be a concern, it may be to say that over the period of evolution, these 4 residues from this subject, that subject is nothing but CCR2, right? This CCR2. These 4 residues got deleted in the process of evolution, or these 4 residues are inserted in human CCR5. Okay, either of the ways, either it is deleted or it is inserted in the human CCR5 when we are comparing only between CCR5 and CCR2. Okay, now any guess from anyone, how can we make a conclusion that it is the insertion that has happened, or is it a deletion that has happened over a process of evolution? Do I need to repeat the question?
So by comparing the
Yes, sir.
Okay. Now question is, this query refers to human CCR5, subject sequence refers to human CCR2. Okay, so this is CCR5 and and this is CCR2. What I mentioned is there are 4 gaps. That means 4 letters are missing in this. When we say 4 letters are not there, that means it is a deleted. Either 4 amino acids got deleted in the CCR2, or this may be the correct sequence 370, but QY, QF, these 4 amino acids got inserted in the process of evolution in human CCR5. Now my question is there any way by which we can say that the insertion happened in CCR5 or deletion happened in the CCR2? On what basis we can make that conclusion? Or can we make such conclusions in this alignment? Yes, it is deleted in CCR2, or no, it is insertion happened in the CCR5.
So, on my views, we can't get any conclusion regarding this because CCR5 is different from CCR2. Then how can we get that? We just comparing the both the protein sectors,
Yes,
so we can't get any alignment.
correct. Now, we cannot make the conclusion whether it is insertion or deletion just by looking into two sequences. Okay. Now, to make any such conclusions to be made, we have to take more sequences that are very closely related to CCR5 and more sequences which are very closely related to CCR2. And then if we compare them, then we will get to know, like, yes, this is how it was in the beginning, in the sense the first protein was CCR5. From CCR5, then the CCR4 existed or derived, then CCR3, then CCR2. So this is how we will also do that particular experiment, orthologs and paralogs. So this is how different proteins are derived during the process of evolution. And in the initial, the number of amino acids, the first set was 374. Then the deletion happened, and from the 374, some amino acids got deleted, and made it like 352, which we say like CCR5. But funny, in the beginning, it has 374 amino acids, but later few amino acids got deleted from that and then attained into 352. To make such conclusion that the process deletion happened, we need to take multiple sequence alignment. We need to do multiple sequence alignment, then we can make those conclusions. So that also we will do in the tomorrow's class. Now, in today's class, we will restrict ourselves to this blast result analysis. Okay, and always remember, this positives includes the identities also. Okay, so that is why the positive value will be always greater than the identities. So if it is 79%, usually 90% or 87% positives will be there. Out of this, we have 4 gaps present. Because of these gaps only, we have the perfect alignment generated. Okay, any doubts with this? How to
Sir?
So,
yes, please.
sir, please elaborate the mismatching as well as with the gaps. Please elaborate a bit more.
Okay, see, the mismatch is different than the gaps. Okay, now in other words, the mismatch is something like, as I given the example, so there is a faculty who is taking the botany subject, but you have asked the zoology faculty to take a class over there. Okay, let us say this is the botany faculty who is taking a class, and then I will take any. Okay, so this one. So just to give you a comparison or analogy to understand. So you have a botany faculty who is taking the class, but you have replaced with a zoology faculty to take a class. Then we call that one as a mismatch. Okay, that is referred as a mismatch. The gaps were introduced. Okay, here I have. The gap says that there was a botany faculty who was taking a class, but you removed the faculty, but you are not replaced with anyone. You have a faculty here, but you are not replaced with any faculty. You have a faculty here, but you removed. So you are not replaced at all. You just removed that faculty. Then we say that one as a gap. So the gaps are more dangerous than the substitutions. Okay, substitution is much better. You can always say like there is a substitution that happened. Okay, so the faculty got replaced, but the replaced faculty is not good enough for that particular course. Okay, so you will say like mismatch. That's a mismatch. The term itself is a mismatch, right? the faculty is not matching to that particular course, so mismatch. But the gap is there was a faculty who is taking the class, but you removed them, so it becomes like a blank. Blank, in other words, gap. You have created a gap in that particular course. You are not replaced with anyone. Then its effect will be more. Now, whenever such things happens, let us take the same example, right? In the Department of Botany, you removed 4 faculty who are taking the class, then you have created the gap in that particular program or in that whole department. Do we need to put some penalty for this? Because you made this mistake. There are 4 members who are taking the class, but you removed them and you have not replaced them. So then do we need to put a penalty for this? Yes, it is a mistake from the chairperson or whoever. So we have to put a penalty for this. That is where exactly the input, I will go back to this, that is where exactly this gap cost refers to. So the gap cost is, though the values are 11 and 1, it is in negative. Cost is a penalty. We say it like a gap penalty. Okay. So the gap penalty is when you made a first gap introduction. Okay. When you made the first gap introduction, we will put a high penalty for that. That is minus 11. What is that minus 11 refers to? That minus 11 refers to whatever the value you have it here, right? If you look into all these things, right, all the values are going up to minus 3 or minus 4 or minus 5, like that. That means the penalty is not that high when you replace a faculty. Okay, you are somehow adjusting with another faculty to take a class. So if it is a completely mismatch, the instructor is not at all matching with any of the requirements, what is the maximum penalty that you can put is minus 4, because still the attempt is made to replace with something. But when you replace and not going to put any faculty, when you replace and not going to put any faculty to that particular course, then the penalty is very high. You will put like minus 11. Okay, so that is the gap opening or existence. And if you continue to do this mistake every time, you will continue with minus 1 extension. Gap existence is opening. Then the plus, this is the minus 11, then minus 1, minus 1, minus 1, it will extend. That's the gap extension, 1, 1, 1. We have different options here. Okay, you can also make it like you can change these penalties based on what you look for. First time when there is a gap introduced, I will put like minus 11, minus 13, but let the extension be like plus minus 1. But if the opening is there, I don't want to put a huge penalty. I want to put like minus 6, but every time that extends, that then I will put like a minus 2. Okay, then extension becomes 2. Now, by changing these values, the alignment is going to change? Any guess? The current cost is minus 11 and minus 1. If I change this into something like, let us say the least value, minus 6 and 2, if I do like this and do the alignment, that's the blast search, whether the alignment, whatever I'm going to get, will change or not.
No, sir.
No, sir.
I think no, sir. But only the value of gaps penalty will change.
Whenever you change these penalties, okay, the alignment is also going to vary. Okay, whenever you change the penalty, this is also going to vary. But of course, it depends on all these conserved amino acids. So in a very layman understanding, okay, so this penalty is something like whatever the police who is going to put a penalty for your mistake. Okay, that is how it is. If the penalty increases, the number of introduction or number of mistakes that is going to happen will reduce. Okay, if the penalty is very less, what we are telling to the tool with these values is, if I increase the penalty, I am telling to the tool that don't introduce the gaps wherever you feel like. Okay, it's something like, if you just remove a faculty from the department, okay, then the department will be given a negative score. Okay, because there is no faculty to handle that course. Then for the department we will put a high penalty value. Then the chairperson will really think whether to remove the faculty from the department or not. If the penalty is very less, and if there is not going to affect the department, then the chairperson will keep deleting the faculty or will keep introducing the faculty. Either substitution, either keep introducing irrespective of requirements, or just keep deleting the faculty because it is not affecting the department. Then the penalty is not playing a role. So that is where the penalty plays an important role. So if you decrease the penalty, right, let us say without helmet 100 rupees is the penalty or 50 rupees is the penalty, more people will start going out without helmet because the penalty is very small. But if the penalty is very high, everybody starts thinking to wear because the penalty fine is so high that better to wear and go. It's exactly the same way. Now, how it affects the entire alignment? Any guess now? If I increase the penalty, more gaps will be introduced or less gaps will be introduced?
Less gaps.
Less gaps, sir.
Less gaps, sir.
Exactly. Now, when the less gaps are introduced, because the penalty is high, less gaps are introduced, your query coverage will increase or decrease?
Decrease.
Query coverage, you have that one here already in the summary table. Description, query coverage. If I increase the penalty, you said like the number of gaps introduced becomes less. When the gaps introduced become less, right? So the query coverage
will be increased.
Increase or decrease?
Increased, sir.
Query coverage. Now, what is this query coverage refers to? This is the query coverage, right? If I increase the gap penalty so high that because it could introduce these 4 gaps, right, from here till here, it has covered. That means from 13 to 306 of my query sequence, it has covered just by introducing these 4 gaps. Now, if I increase the penalty, it may stop here itself. Okay, it will stop here itself because the penalty is so high that the tool does not want to introduce any gaps. So it will end up here. Then how many residues are going to be covered? It is not 306. It will be around 250 or 200 plus. Then the query coverage will decrease. Okay, so the query coverage will decrease if you increase the gap penalty. Clear? So you can look into the different alignments. Okay, we have different alignments that are being present here. We have like 43% identity, 61% positives. There are 5 gaps introduced. Now, is those 5 gaps continuous? No, there is one here. Then this region is matching, Q, K, E. Now, I have to remember this table. Q refers to glutamine, K refers to lysine, E refers to glutamic acid. So this is mandatory. Okay, so for most of the amino acids, it is easy to remember. There are only few. like R for the arginine, because A is given for the alanine. So though the letter starts with A, then as per gene, N it ends with as per gene, so N is given. Then the D is aspartic acid, E is glutamic acid. And the way you can remember this is D letter starts first in the A, B, C, D, E, right? So A starts first in the aspartic acid. These are the two acidic amino acids. Then G is the next one after A, so glutamic acid. So let's give E, which is D and E. D is A and E is G. So glutamic acid. That's how I remember the amino acids. I first remember that D and E are the acidic amino acid. D, because A is the atomic aspartic acid, A is the first, so let's D is the aspartic acid. Glutamic acid, after A the G comes, so E is glutamic acid. Glycine, histidine, isoleucine, phenylalanine. So pronunciation goes like phenylalanine, that's why F. Okay. Then tryptophan, so unique W. Y is here, tyrosine, Y is tyrosine. So if you remember with this, then interpretation or analysis becomes easy. Phenylalanine to leucine, not the lysine. K is lysine. So that is how plus. Later we will look into now whether this is a good alignment or a bad alignment. That all depends on your project. Okay, what you want to do. Here if you look, there are more gaps. There are 12 gaps being present. So if you keep scrolling down, so we will have the distinct protein. That means compared to CCR5, okay, if we come back to this table, the values keep decreasing. More gaps are introduced, your score gets reduced. Total score gets reduced. Exact match is 373. Then the next match is having a huge variation, 475. So there is a huge difference between CCR5 and CCR2, but 83% query coverage, and within this 83% of the query coverage, that query coverage you can understand it here. This is my 352 amino acids, and this is where it is matching. First one CCR5 to CCR5, but the next one is starting from this particular position 13 or 32 and ends up to here near 305. Then the next hit is from here to here. So is there any entry which covers the entire 352 amino acids, right? This is 350 and then 2. Is there any which covers the entire 352? No. There are some regions which is not matching. These are poor. Accordingly, the coloring scheme is, I think the coloring scheme is also given. The coloring scheme is based on the score. That's where it is. If the score is greater than 200, red, pink, green, blue, black. So if we have like anything in the black, that means they are not matching at all. Green we have. So the green refers to 50 to 80. So if you click on any one of these alignments, you will get to know. So the score is 79.3. That is why it is given into 79, right? So in the pink color. Why it is 79? Because there are so many gaps. So for any gap when we introduce, we subtract the value. Okay, when we have positive, we will add the value. Positive is we have positive values. All the positive values gives a good alignment. All the substitutions if you make, negative values. And if you introduce a gap, that means you remove the faculty, higher penalty. So for that, the score becomes very less. Any doubts with this one? Now let's look into another value, okay, even which the professor was mentioning, that is the E value. Now can anyone explain me or tell me what exactly this expectation value refers to, or this E value refers to? Just this is the first alignment, CCR5 to CCR5. and E value here it shows like 0.0. Then the CCR5 to CCR2, the E value is 4E minus 169. So any guess or your understanding about this E value? Why we have like 0.0 here? And what is this 4E? This 4E minus 169 is refers to 4 into 10 to the power of 1 minus 169. In other words, 0.000000169 zeros and then 4. Okay, so 169 zeros and then 4. 0.000004. So the tool says like it is not still 0. Okay, this 0.0000169 zeros and 4, it says like it is not equal to 0. That's what the accuracy of this particular blast search is. Now, what is the difference? Some guess. Expectation value, morning it was just mentioned. You can just make some attempt to explain what it is.
Significant value.
Correct. Significance value.
Sir, E value will tell you about the homologous sequences, how much both the query as well as the target is matching.
Correct.
The above target, which is showing 7 to 3 bits, they are the same. So that's why the E value is 0.0. And the next one is CCR2, which is bit different than the CCR5. So that's why the value is higher, I think so.
Correct. Some more ways by which even a layman can understand this one. Any ways by which someone can put this one, because it's still too difficult to understand this, right? What is that expectation, and what is there any threshold, or what is that 0.0 refers to? Any what is this 0.0 refers to in the expect value? Any attempt? All are correct answers only. What is that 0 refers to? 0.0 or 0, both are one and the same. What is this 0? Yes, sir.
The sequence is having 352 amino acids and all are covered and all are matched. There is no gap, no penalty. So it is 0.0.
Okay. Now,
there is no gap.
Correct. Now, actually, the way it works, or what we should understand from this E value is, it is not saying that there are 0 gaps or 100% match. This expect refers to expectation value. Okay. So, please listen to this one. This very, very challenging or difficult concept to be explained to any layman or to whomsoever. And we get this expectation value in most of the algorithms or in most of the bioinformatics or even any other techniques that we use. Now I will just make an attempt to make this whole concept easy to understand. Now, expectation value refers to who is expecting. The first question is the, let us say, the customer or a person, whoever searching, right? I am searching my query sequence within the database. Now let us put this into a very simple or a analogical way of understanding. Okay, analogy is something like you want to buy a saree, okay, and you will go to a shop, and then the shop person is going to show you one particular saree. Then next question, you are expecting that saree. Okay, now the expectation is something like, your next question is there any other saree which is exactly like this saree? Let us say you want to have 2 identical sarees, maybe to give it to some your sisters or whatever, for example. Now the shopkeeper is going to say like, I do not have any other saree which is matching to this. Okay, this is the unique piece available. You cannot find the same quality or the coloring, the pattern, whatever is there. We will not find the exact saree. This is the only saree available. Then the saree person can tell this one. This is the unique piece. Now, how can a tool is going to tell this, right? So the tool, because the saree he can tell because he knows what is there in his store. Okay, you cannot tell like there is no other saree present in the shop. The shopkeeper has to tell that answer. There is no other saree present. Just like the blast knows what is there in its database. Okay, now the value 0 refers to you cannot expect one more alignment which is matching with the exact score. Okay, you cannot expect another alignment which is having the exact score like 723. If there is one more alignment with the 723 or exact alignment matching like this, okay, then what exact? To bring it back to the shop, right? If the shopkeeper has one more saree exactly matching to the one that you are interested, then he will say like, yes, there is one more saree that is there. Then he let give the value like 1. If the value is expectation value is 1, that means there is one more saree which is exactly matching to this. What if the value is 10? Then you have 10 identical sarees. Okay, whether it is required? Yes, if you want to give 10 exact sarees to 10 members, right? Let us say you have 5 staff and you want to have the uniform color code, then the expectation value has to be high. So the shopkeeper will ask, I want 10 exact copies to be there. Then the expectation value will increase. Okay, so it goes with the requirement. Okay, then 5 becomes a good number, 10 becomes a good number because 5 identical sarees you want to give it to 10 members, not to have any conflict. But here in our sequence alignment, if the value becomes 0, that means you can expect only one such alignment to be there. You cannot expect one more alignment from the database. What if the value is something like this, 4e minus 169? That means the tool is saying, from my side, I am accurate. You can expect one more alignment with this probability. Okay, this is the probability value. That is 0.0000169 zeros and then 4, which we can treat it as close to 0. The tool is not going to select 0. This is almost like that much. Then how about this one? This is 169. This is 143. The score is 475. Here the score is less, 409. So with the decrease in the score, there is a decrease in the E value also. Okay, with the decrease in the score, there is a decrease in the E value. Now again, if you want to put that one into a simple perspective, so if you increase the word size, or if your query coverage increases, your score will increase. The finding the exact match is going to be high. So finding the exact value is E value should be 0. Okay, if the E value is 0, that means that's the unique alignment. If the E value keep increasing, that means you have a chance of finding another alignment matching to this. That means there is somewhere the redundancy is there. Redundancy in the means the same protein can be present in any other organism or in any other form also. So if we have like expectation value something like 1, why we are having like 0.00? Because we have so much of data that is being present, billions of sequences. I think that statistics also we should be able to get here. So the search, database updation, and then this is what the Karlin actual statistics. From this only the E value is calculated, and it has checked somewhere. We will get search summary. How many sequences it has searched? Okay, number of sequences that it has searched is 4,77,327 sequences. Okay, my 352 amino acids is searched with so many sequences, and each sequence may have around, or again, 300 amino acids. So that also we should be able to get how many characters it has searched. So when it has searched so much, there is a probability that it is going to match. Now, just to make sure that you have understood the e-value correctly or not, my simple question is, what if my query sequence, this is my query sequence, right? 352 amino acids. What if I submit only 10 amino acids and delete all 352 and submit the first 10 amino acids and search without doing experiment? Okay, just guess it out. More or less in most of the things you just need to imagine. When I give 10 amino acids and do a search in the BLAST with a Swiss prot, can I expect the e-value to be 0, or the e-value can be of higher number?
Higher numbers.
Why? Why? Yes. What made you to say like the value?
Sir, the search area or the number of sequences will match with the multiple things, because the search area is reduced.
Perfect.
That's my
Exactly. Correct. That's where in this search, this word size plays an important role. Okay. If I decrease, what I am doing is, my name is like Prashantha Karunakar. If I just type one letter like P and search in the Google, right, the chances of this P getting matched is so many, 16 lakh, whatever it is. Now, if I increase to PR, the number this gets reduced. If I keep increasing the word size, this is what the word size refers to, PRA. Now, PRA it is not just matching with the Prashantha, right? PRA healthcare or health sciences, there are so many matches that is going to be there with this PRA. Then if I add another one, S, and search, the number keeps getting reduced. H, another letter again, right? So the new thing will keep adding. So when I keep adding just with the Prashantha again as a search, right, it becomes like 2,65,000. That is the matching that is there. There are other stuffs like other names, Prashantha CS, Prashantha CN, Prashantha Upunda, whatever. But if I increase the word size, that means the query coverage I have increased, then the chances of exact match from that 65,000 to it has come down to 18,000. Okay, so the chances of exact matching. What if it is not showing the exact match whatever I'm looking, then I will increase this into PES. What if there are two Prashanthas present in different PES, right? Then increase it. Again, the keywords, biotechnology. What if within the biotechnology there are two Prashanthas being present? You again increase the keywords, bioinformatics, right? Then it will, now if you look into, it has come down to like 449 results. Exactly matching. What if within PES, biotechnology, bioinformatics, there are two Prashantha Karunakars if it is present, right? Still add one more with PhD, assuming that other person may not have a PhD, right? Then with the PhD, it has come down to 292. So what we have done is, if you convert this into our amino acid sequence, okay, if I convert this into our amino acid sequence, with the increase in the amino acid sequence length, the accuracy of getting the exact match, that means having with the 0 expectation value, you cannot expect one more person. What we are telling here is, you cannot expect one more person like the Prashantha Karunakar with this. So then the e-value is 0. If my keywords are less, so the tool is going to give that you can expect one more person with the exact name having a Prashantha Karunakar. Okay, there may be one more person with the same name, because here the Google is smart enough to detect and do it. Here it's all ETGC and we are not giving any constant, so it will just show like there is one more person with the same name being present. Is it clear with the expectation value concept? Or any doubts with this?
Yes, sir, clear.
Okay. Now, if you want, we can take a break, maybe, or if you want, we will continue with the first talk. If you want, now it is 12:57, maybe we can continue. We can take a 10 minutes break, and maybe around 1:10 to 2, we will do another session. Is it fine?
Okay, sir. Okay.
Okay, sir.
Just a 10 minutes break, and we will continue from 1:10, and then look into the first and its alignment. Please don't exit from the group, or because be in this session itself. The session is going to be continuous, because when you come back, right, again the host has to admit you, so that may delay because the host is also listening, so it may take some time, or you may be waiting room. So be in this lobby, okay, so that you can continue to listen. So if you exit, you will be under waiting list.
Mm.
we start, sir?
Yes, sir, we can start.
Four more members to join. 35.
Let us see.
Okay. So, any doubts with whatever we discussed? How many participants? Okay, so we'll continue. Now we will look into the FASTA search algorithm. Okay, as even in the morning class that was mentioned. So it is just referred as FASTA. Okay, that's how it is usually pronounced as. Now the BLAST or FASTA, right? So both are sequence similarity search engines. When we are doing this, just a second. Okay, sorry for that. So both BLAST or FASTA is going to work on the sequence similarity search only. There is a small difference in the algorithm wise, the way it is going to search. So that was also covered in the morning theoretical session, how exactly the search is going to work in both the ways. BLAST is both BLAST or FASTA is a heuristic method, but BLAST looks for the word size in the center and then works out in both the directions. But FASTA initially starts with the dot plot and then goes into dynamic programming and then shows the result. But in principle, when we look into, more or less the results comes out to be the same. Not much of a difference will be there, but with reference to the speed, BLAST gives the results much faster than the FASTA. Okay, now let us do with the same sequence. Okay, just type the FASTA. Then here also, just like in the BLAST, we have BLAST P, BLAST N, T BLAST N, T BLAST X. So here also we have the same kind of aspect. Now, in the first instance, there also we have the database to select. So here also we will select the same. We have option to select multiple databases here. That's one advantage. And if we want to search within the Tremble, okay, we can select that automatically annotated section. So we will continue to do with the same UniProt, SwissProt. Okay, then submit the sequence here. Go back to human CCR5 in the UniProt, then get the FASTA sequence, then take this particular sequence, entire 352 amino acids, and then go to the FASTA, select the UniProt, SwissProt, okay, and then paste the sequence here, and then keep other parameters as it is. Okay, if you want a specific database, you can also change UniProt clusters if you want RefSec anything, or if you want to search in the PDB, we can select this. So we will use this option of selecting the PDB. Even we have an option here to select the PDB. This we will do when we do for the sequence structure similarity search at that particular experiment. Okay, as of now we are working out at the sequence level, so not to worry about the structures. So in the sequence level, still we are interested in the matching of the human CCR5 with other organisms, and we will select the UniProt, SwissProt, keep everything as it is. If we have a sequence to upload, you can select and upload the sequence. Since we have option to paste, we will paste it here, keep everything as it is. Here also the more option, right? So as we discussed, we have like gap open and gap extension penalty. So since it is a penalty, the values will be negative. Okay, so here the penalty can go from by default, which is -10, we can go up to -64. Okay, so here we will work out with by changing these penalties what happens to the alignment. Okay, so it becomes very easy. The default matrix BLOSUM 50 and then other PAM matrix is also there of BLOSUM. Okay, let's keep everything default. BLOSUM 50, gap opening penalty -10, gap extension penalty -2, word size or the k tuple 2, expectation value we want from the 0. Now we know the what exactly expectation refers to. So the unique one, 2, even if there is some overlap in the values or another value, another alignment matching to the one it has given. So we are going from 0 to 10. Okay, so usually the expectation will not be negative. Okay, but 0 is the one, 0 is the unique one. Then whether it's a DNA strand or if it is a DNA, then we need to take what. So it is not applicable. So don't change anything. Let it show 50 alignments. Then the just like the sequence range, if we want to give anything, we can give. If we are not providing anything, it will take the entire 352 amino acids. High segment pairs, whether it has to show, don't do anything, keep everything default, and then just say submit. Okay, now what it is doing is it is searching our 352 amino acids with the entire SwissProt database using the FASTA algorithm. Okay, then we have the results obtained here. Now once again, our 352 amino acids of the query sequence is matching with the 352 of the P51681, which is the same 352. Now the score is different here. Whatever the score that we obtained here for the P51681, the bit score was 723, but while when we do with the FASTA, the score is 495.4. Okay, any guess why it is different? Because the BLOSUM matrix that we used is different. Okay, now if you recall with this, the matrix values of this will change from BLOSUM 62 to BLOSUM 80 or BLOSUM 90, like that. I will also discuss the difference between the PAM and BLOSUM. Okay, so that you will know when should we use spam, when should we use blossom. That also we will understand today. 100% identity, we know, yes, 352 to 352 exactly matching, and the percentage positives is also 100. Here it is not showing like 0, okay, but rather it is showing like 5.3 E minus 139. That means 1 into 10 to the power of 5.3 into 10 to the power of minus 139. In other words, 0.00053 after 139 zeros. So that means there is still someone, but some more alignment. The E value is this much, but we can take it as 0. It is much more critical in giving the E value compared to BLAST. Okay. And now if we want to see the alignment, okay, so the first 50 it is going to show as a result here. And if we want to see the alignment, so we can, we have an option here itself, okay, show alignment. Okay, alignment show. If we click this one, now here we will have the entire alignment. So rather than placing, now you can maybe spend some time and can you tell me the difference in the alignment between the BLAST result and then the FASTA result? What is the difference in the representation, or what are the additional information that you are getting in the FASTA when compared to BLAST? You can compare, maybe you can keep the two windows and then you can make a comparison. Okay, this is with the FASTA and you have a result from the BLAST here. Maybe you can place it here, then make it like a 2 screens, and then you can do the comparison. Yeah.
Sir, in BLAST they are telling that query and subject, but here there is a something number called SP.
Correct.
And also we are having the 10, 20, 30, that base, I think that is a base pass number.
Yes.
But here there is no different base pass number like that.
Okay.
Past.
Here we have. But the
I think,
not
yeah.
the 10 interval, but 60 residue interval is given here. But here for every 10th position, we have the numbering. Very good. Correct. Then
the middle line.
Yes. Perfect.
Some letters are there. It is here some dots are there.
Perfect. Correct, sir. Now, whenever we have like a colon, so that refers to the identity. So wherever the identity is there, rather than giving the same text, here the symbols are used. Good observation. Then some more, maybe if we want now, let us look into this one. That becomes, maybe if we want, I will zoom in a bit more. Okay. And look for this alignment. For
two sequences are shown with their identity number. Yeah.
Here in the BLAST.
No, in FASTA.
In FASTA. Identity, this percentage.
No. SP P51, yeah. SP P60.
Yes, this is the Swiss prot.
Yeah.
P51681 is the human CCR5. This is P60574. That is the CCR5 from the Paniscus, probably a monkey, okay, organism. So if you want, you can just right click and search. Should be a monkey. We'll get that image. Balloon. Okay. Chimpanzee. This is. Yeah. Good. The taxation number is what is given here. Then some more. Is the symbols are the same? We have something like a dot here now. What does this dot represents now?
It represents mismatch.
Dot represents the mismatch.
I think, sir, it may be a gap.
Think it may be a gap. One mismatch, one is gap. What is this dot represents? We have N and D. N and D.
This is a mismatch.
Okay. Then we'll take one more example where we have a dot. Do this. To get
Similarities.
yeah. So let us look into another alignment where we have again N and so now here no symbols are there, but we have a single dot here. We have a single dot here. I and V. I refers to isoleucine. V refers to valine. Now isoleucine and valine belongs to, from the chart, if we recall, hydrophobic and aliphatic, right, amino acid. So that refers to similarity. That means the dot refers to similarity. And if we have a colon, that refers to identity. Now, if nothing is there, what does it mean? It's the same as this situation. Mismatch. Okay. Now, comparing with the blast and this, if we have a colon, that is identity. If we have a single dot, that's the similarity. Whether you say similarity or positive, both are one and the same. And if there is nothing, that means it's a mismatch. Now, how do they represent the gaps? Gap represents in the same manner, whatever we have it in this. So if there is any gap, that is in minus symbol. Just need to check out where we have any gaps introduced. Yeah. Okay, so this is the representation for the gaps, which is same as whatever we observed in the blast result. So if there is any region where it has to introduce the gap, that remains the same. Minus symbol for the gaps. Mismatch, nothing they are going to put. Then if it is a conserved, then a colon. If it is a single dot, positive. Okay, that's how it is going to be, the comparison. Now, if we look further into the results, we have different more statistical values coming out in the FASTA compared to BLAST. We have like init-N and init-1. Probably even in the morning class also it was mentioned. So to let us not go into all the details, but I will just put you a perspective how to analyze this. If the init-1 and init-N refers to like how many fragments are making this alignment. If there is only one fragment, one sequence, one complete sequence, if it is going to match, then we have init 1 and init n matching. So if there are two fragments that is going to make, maybe I will show you this init 1 and init n here. Sequence match and the subject match. Sequence match was our query sequence. Subject match is from the database. If one fragment is sequence matches with the subject match, if we look into complete 352 amino acids, this one, the end is 354, it is showing, but the length is 352. So the region which is matching, if there are two fragments coming out, then the init n and init 1 values is going to be varying. Otherwise, both the values remains the same. Okay, so it goes a bit complicated, but nevertheless, so now just check out where we will have the gaps. Okay, and just tell me that particular alignment where we have the values, where in which particular accession number we have more gaps being present. You can just tell me that accession number. Check out, scroll down, and then tell me which alignment has more gaps being present. Yeah, could find out the accession where. So you are also getting the same result, right? Just need to enable this alignment. Then you got to keep scrolling. Is it like the same two gaps that you are also obtaining? Now, if there are only two gaps that we are obtaining here, even in your alignment, because we have given like 50 results to be shown in our input page. Okay, so our input page, in the options, it was 50. So that is why it is showing. If we increase it to like 250 or 500,000, we will have more gaps to be. Let us say we will retain with the 50. Okay, now if I want to see more gaps in my alignment, what I should do?
Extend the gap.
Extend the gap. So should I increase the gap value or decrease the gap value? Now the default value is -10. Should I go to -20 or should I go to -1?
-1.
-1? How many of you agree with this? You can quickly raise your hands, or that means we will get to know. It's slightly complicated or difficult at the first instance, I do agree, but it's quite interesting the way it works. And now you can also tell me what are the changes to be made. Now, how many of you agree that it has to be -1? So if I decrease the gap penalty, more gaps will be introduced. And one more important aspect is your gap opening penalty, okay, your gap extension penalty cannot be greater than gap opening penalty. Okay, if I am having like -1, this should be -1 at least, or can be like 0. You cannot have the gap opening penalty -1 and gap extension penalty like -5 or -6. Okay, so if we keep both the penalties same and ask it to search, then the more gaps will be introduced because the penalty is very less. Okay, if I don't want even a single gap to be introduced, then increase the gap penalty to -50 or -60 like that. So increase the penalty so that we will not have any gaps. Now, furthermore, to our exercise, basic question comes out: on what basis we have to fix this gap, right? When I should increase the gap penalty and when I should decrease the gap penalty. When the default is -10, when should we take a decision to decrease the penalty from -10 to -1, or when I should take a decision that I should change from -10 to -20 or -30? When we should do that? Any guess? Any guess from anyone? Do I need to repeat the question?
Yes, sir.
Yes, sir.
Now by the default value of the gap penalty is -10, right? When we should take a decision to reduce the gap penalty from -10 to -1, or can you tell me a situation when we should increase the gap penalty from -10 to -50, for example? When we should think of changing the gap penalty values is what I'm asking.
To increase the number of gaps or to decrease the number of gaps in the sequences, sir?
Correct. Now, what made us to think of increase the gap, or when should we think of not to introduce the gap?
To cover the coverage, on the basis of the coverage,
Yes.
gaps should be introduced, more gaps to cover more the sequence. If we the gaps or reduce the gaps, the query coverage would be lesser.
Perfect. Correct. If I'm finding like, if I look into the last alignment, right, and if the coverage is not having the entire thing, here in this case it is matching. Let us say in your real-time experiment, my query sequence is 352 amino acids, but if the result whatever it is showing with the default -10 as a gap opening penalty and -1 or -2 as a gap extension penalty, but in the result, if it is not covering entire 352 amino acids, but if it is covering only like, let us say 250 amino acids, okay, then we will get to know like, because the gap penalty is -10, okay, it is not introducing the gaps to cover all 352 amino acids, because the penalty is -10 and we always give a threshold. Okay, there is a cutoff with a threshold value. Anything which is less than, don't include. To put that one back into our perspective, okay, for our easy understanding, the gap is a penalty. Okay, again, if the penalty value is so high, okay, if we have put, again, if we come back to the penalty as as a percentage. Okay, now you want some candidates to come for the interview. Okay, but the penalty, or if you have put so much of criteria, and in the criteria that you are placing, there is no application coming for the interview. Okay, then what will you do is you will decrease the penalty. That means you will decrease the penalty so that at least you want to cover some candidates for the interview. So in the same way, if I decrease the gap penalty, your query coverage will increase, because you are allowing it to introduce the gaps wherever it is possible and to increase the query coverage. So in that situation, if your query coverage is getting reduced, then decrease the gap penalty so that the more coverage will be there. Then you want to see where and all it is matching. So then that's where the gap penalty plays an important role. In other cases like this, anyway, the 352 amino acids are covering even in the 50th. If I look into even in the 50th result, if I look 352 amino acids starting from first amino acid till the 352 amino acids, entire thing is matching. So decreasing the gap penalty will not give me the closely related. So I will get a hint that increase the gap penalty. So even if I increase it to -50, the query coverage is still there. So I will get a more closely related. Now, is there a better plan or better way people thought of? Okay, because this gap penalty is making too much of mess. Gap opening with -10 or gap opening with gap extension with -2 or -1, not affecting too much with alignment. Is there any other way by which I want to find out? Now, let me tell you how the analysis should be. Okay, not getting, just getting these results is not important. Okay, what is that closely related is what we finally look for. I have a human CCR5 and I want to look for closely related sequence. Okay, so what I am getting is with the default blossom 62 or blossom 80. I have human CCR5 and human CCR5 is matching. That is fine. The next one what we got is this, that is the chimpanzee that we obtained. Then monkeys or gorilla, right? These things are coming. And now if I look into this ratus, this 50th hit, and if I look for this, which organism it is, belongs to like now the brown rat. Okay, now I have a ways starting from human to gorilla to monkeys, right, and then coming till the rat, that is the 50th one. Now I will get these results if I am using with this blossom 50. Now even in the morning class, right, it was mentioned with two matrices, blossom and then the PAM. So let's work out the difference between these two. Blossom refers to block substitution matrix. Okay, maybe just to give you the perspective of this, I will just give you one. Oh, they are related to each other. Or any, because it's already done in the morning, can you just tell me the difference between PAM and blossom? Someone? Can you make some attempt in explaining the difference? Meantime, I will find out the difference between the PAM and blossom. Okay. Any guess? We'll make it blank. Any guess?
Sir, my guess is to the PAM is point accepted matrix. That is, we can correlate with the exact point or the closely related point. Whereas the BLOSSOM is block substitution matrix. It may be a group which the matrix or the point which defines as a particular group.
Correct. Good. Now the PAM refers to, I don't know whether this PAM is not there. PAM refers to point accepted mutation. Okay, not the matrix. PAM
Okay,
is point accepted mutation. Maybe I will just open that one also. Quickly you can understand. Okay, so PAM refers to point accepted mutation. Now let's not go into all these slides because it makes life come a bit difficult. Just quick understanding. PAM 1, okay, we have PAM numbers given or BLOSSOM numbers given. If you look into PAM 250, PAM 120 like this, okay, don't look into other tools or other algorithm at this point of time. Now PAM 1 refers to one mutation out of 100 residues. Okay, the term also important. Residues, we use the term residues whenever we are working with amino acids. Whenever we are working with nucleotides, we call it like 100 base pairs or 100 nucleotides or 100 amino acids for the proteins or 100 residues for the amino acids. If it is a nucleotide sequence, it is referred as 100 base pairs or 100 nucleotides. Okay, that's the first thing. You need to be very clear with the terminologies. Even if you observe right in the morning session, so the professor has clearly mentioned it is not the protein-protein interaction, it is the protein-protein comparison, because the interaction is different, comparison is different. So in the same way, residue, if we use the term, we get to know that it is with amino acid. Now PAM 1 refers to 1% of mutation is accepted. That means out of 100 amino acids, one amino acid change, if it is there, then that is referred as PAM 1. PAM 50 refers to 50% of the residues, that is 50 amino acids out of 100 nucleotides. If it is changed, it is accepted. That is point accepted mutation. That mutation is accepted in the evolutionary process. Okay, that is what the accepted refers to. It is not like mutation happened and the organism becomes extinct. Then such steps are not accepted. So the mutation should be accepted by the nature. That means the organisms that are surviving now is considered to be the accepted mutations. If the mutation happened and the organism is not surviving, then that is not a good mutation, because that is not supporting the organism to survive. So such mutations will not be considered. Now PAM 250 refers to 250 mutations out of 100 amino acids. Okay, how is it possible that 150 mutations out of 100 amino acids, or let's say PAM 250 mutations out of 100 amino acids, right? So the concept goes something like this. One amino acid to another amino acid is not going to happen in a straight manner. So there will be an intermediate amino acid changes that is going to happen. Then it will retain there. So in other words, to put this one into another easiest perspective, so always the mutation going to happen with the timeline. Okay, if there are 100, let's say 1 million years, or just to put it into another simplistic manner, so there is a bus, okay, that is traveling from here, let's say from PES to Majestic in the Bangalore, which is going to travel for 1 hour. So the time is 1 hour. Is it possible? There are 100 seats in the bus. Okay. Now, out of 100 seats, is it possible in the process of 1 hour, 250 members to be present in the bus? Is it possible? Yes. because the guy will get on intermediate for every 15 minutes, 30 minutes, or one guy may be sitting from first minute to the 60th minute. That guy is referred as conserved. Okay, and there is a possibility that the amino acids, that is the members, get down and in the same stop another member getting in, right? Boarding, deboarding, boarding, deboarding happening. So at the count, when we take 250 tickets were borrowed, that means in 1 hour 250 tickets are borrowed. That is referred as PAM 250. What do I mean by PAM 1? That means from first stop 1 minute to the 60th minute, 99 members are conserved. That means they will get on only at the last stop. But there is only one guy who is changing, who will get down and another member will get in. One difference is allowed. That is referred as PAM 1. I hope it is clear, right? Analogy you could understand? PAM 1 and PAM 250. Or do I need to repeat?
Sir, can you please repeat?
Repeat, sir.
Okay. So again, just to make this concept to understand, these are quite interesting concepts. Okay, but okay, so let's say the mutation, okay, point accepted mutation it is. So usually the mutation takes huge amount of time, specific to humans. Now always the mutation happens with reference to time. So to change the perspective of this amino acid from alanine to methionine, methionine to histidine, that change, that is slightly difficult to understand. Because so what I thought is to put another perspective. Let us say you have a stop from one point to another point, which may take, for example, 10 stops to reach here. Okay, 10 stops to reach here. We have different stops that are going to be there. Even you can imagine your own city. From your home to your destination college, you may have like multiple stops that are there in the city bus. Now, from here to here, when you are traveling, let us say in your bus you have 100 seats available, because PAM is percentage, 100 seats available. If you start from the first stop and when you get down here, let us assume like in your bus you have 100 seats filled and 99 members if they get down here, okay, that is like your college bus. 99 members will get down here and one person may get down somewhere else and another person may get in here at some particular point of time. One will get down and another, that is a substitution that has happened. One guy get down and another get down. 99% is conserved. All 99 members are here. That means this particular situation is referred as PAM 1. This 1 refers to only one mutation, only one change is allowed out of 100. Out of 100, only 1% or one person is allowed to change. In other words, this is refers to the protein. If we take the protein as example, the protein throughout the evolutionary period, no mutation happened. Only one mutation is happened. If such you want to identify, if such protein where 99% identity should be there, then we go with the PAM 1, which is that bus which is taking 99 members and allowing only one member to change, which is that bus, which is that protein. Bus is nothing but the protein now. What if I want another bus where it should allow me to go for multiple entry and multiple exit? And then if I give like, let's say for example, PAM 100. Okay, if I say like PAM 100, that means at the end of the journey, it's all at the end of the journey, there are 100 changes that are happened. That means 100 peoples, 100 members. Someone will get down and someone will get in, get out, okay, or deboard, on board, deboard, on board, deboard, on board. 100 members. That means the conductor has issued 100 tickets. Okay, then we want PAM 100. You want another situation where there are so many entry and exit happening. Well, then we call that one as PAM 250. That PAM 250 refers to the conductor has to issue 250 tickets within the same journey. So when is that possible? That means for every stop, right? For every stop, there are few members will get down because the space should be there in the bus. Okay, don't imagine too many things standing and all those things. This is just for analogy. Okay, then again, there are few members will get down, few members will get in at the same point. Mutation happened. Again, few members will get down. Again, few members will get in here. So at the end of this journey, he has issued 250 tickets. Now, when this happens, 250 tickets are issued in this time frame of this 1 hour. Okay, let us say the 60 minutes of travel, 1 hour journey, 250 members getting down, and that means there are so many substitutions that are being happening in this. So many substitutions are happening in this. Now, when there are many substitutions, what do you call it as? Difference. So whenever if it is 99% identity, that means they are very closely related. Okay, that is highly conserved. This particular bus is conservative, highly conserved. Once he starts, he will not stop. He will carry entire thing and stop there only. But this guy, this bus has too many flexibilities. Okay, this has too many flexibilities. Whenever they want, they can get on, and there are some members getting, and then there are too many flexibilities that are there. That means this protein, whatever we get, or this bus, we call it as a flexible, and this bus we call it as a rigid or conservative, conserved bus and flexible bus. Is it clear now? Now, how is this related with our protein? Now, I have 352 amino acids, right? And I want to see another protein having too many variations compared to my protein. Okay, I want to see another protein which is having too many substitutions with my protein. Then which one I should use? PAM 1 or PAM 250?
PAM 250.
Perfect. So if I select PAM 250, I am asking the tool to show me that bus which allows me to have too many flexibilities. If I do not want flexibilities, okay, then we will go with this PAM 120 or other things. Now coming to this Blossom, okay, Blossom is another tool or another algorithm which works something like, again, if I bring back this, that is lost. If I want to bring back this here, Blossom is block substitution matrix. Now, higher the number of Blossom, okay, let's look into this. PAM 0, that means 0 mutations observed, 100% identity. PAM 1, 1 substitution allowed, or 99% identity between two proteins. PAM 250, 80% mutations are observed, that means 20% identity. While the Blossom is opposite, higher the Blossom number, okay, higher the Blossom number, more will be the identity. Lesser the Blossom number, lesser will be the identity. If I say like Blossom 62, that means, if I say Blossom 62, it means I am looking for this BLAST P62 or Blossom 62 are one and the same, because the default program in the BLAST is Blossom 62. That is why they have given like BLAST P62. 62% identity should be there. Then we say like Blossom 62. 80% identity should be there between two. Then I will go for Blossom 80. Okay, so that is how both are interconnected. So if I have to show you the correlation between these two, okay, so PAM 100 is nothing but Blossom 90. If you look into increase in the PAM value, there is a decrease in the Blossom value. PAM 250 is nothing but Blossom 45. PAM 200 is nothing but Blossom 52. PAM tells about the more mutation. Blossom tells about more block substitution matrix. This 52 tells the identity. 90 tells 90% identity. 80 tells 80% identity. Now 80% identity is nothing but 80% should be same, 20% difference can be allowed. Okay, so when you are decreasing the difference, more similarity you are looking for. Now you can also do this quick experiment. Change it to PAM 250. Don't change anything. Keep everything same. Increase this to PAM 250 and then perform the faster search and then check. Not the first one. Okay, because first one is always, it looks for giving the accurate, closely related. P51681 or anything. But look for the 50th one. What will we get here? And whether there is any change in the result, the score, the alignment, all those things. You can just check how is the result. If it is still the same, okay, if it's still the same, that means the even with the change in the matrix, the closely related sequence, there still remains to be in the close set of organisms. So then we will change with the gap penalty. So these two are the major parameters, gap penalty and then the matrix are the two major parameters by which we work out in identifying the closely related organisms and then the far related organisms. What we obtained there and what we are obtaining here, just check with that accession number, score, whether it is going to be the same or different. So in our typical class, okay, in our undergraduate classes, so what I usually do is we will keep changing these parameters from PAM1 to whatever the options we have, all possible options, we will keep changing one parameter at a time. That's why even in your list of experiments given parameters affecting the alignment, you keep changing this from PAM 120. Look at the alignment, how it is. Then change it into PAM 250. Look at the alignment, score, everything. How is the difference? Then keep the PAM 250 and increase the gap penalty. Okay, how is the result? So that comparison, once you start doing, then you will get the correlation between these two. So how the alignment is going to affect or the results is going to affect when I increase the gap penalty, decrease the gap penalty, increase the PAM value, decrease the PAM value, decrease the Blossom value or increase the Blossom value, how they are going to work. So that linkage you are going to get. So what is the first result? What is the 10th result, 20th result, 30th hit result, 50th hit result? Which organism, what is the score, how is the alignment? Is there any gaps introduced or not? So overall, when you do all these exercise, your conclusion will be something like this. When I increase the PAM value, far related sequence we will get. This becomes like far related organism, not a closely related. When I decrease the PAM value, I will get the closely related organism. Blossom also, that will be your conclusion. When I increase the Blossom value, I will get a closely related organism. When I decrease the Blossom value, I will get a far related organism because Blossom 10 refers to 10% identity. Then gap value, if I increase the gap penalty like -60, So the alignment becomes very short, right? And then the query coverage will decrease, and also I will get the far related because query coverage becomes very short. If I decrease the the gap penalty from -50 to -1, the query coverage will increase, but the organism, whether the far related organism, I am going to cover. So that linkage between these three parameters you will get to know by playing with these parameters. Now, on what basis should we decide? That will be the next question. When I should increase the PAM or decrease, when should I do all those things? So that depends on what kind of experiment you are doing. Okay, so if it is like model organism you want to test, in your laboratory there is a particular organism available to test, then in such situation, when you do the FASTA search, check in this particular list where it is lying, whether it is coming under a closely related organism or a far related organism. So if it is a far related organism, then the chances of success in your experiment is less. But if your organism is very closely related, then the chances of your experimental animal testing when you do is more accurate. So in the same way it is going to happen in the human, that you can make a decision. Okay, so that's how the parameters are linked. That's how we should use these tools or servers, or should play with these parameters. Any doubts with anything? Also, there was one more question in the morning, that is protein comparison. Why should we do when we can do with the DNA or RNA, from one of the participants? Because the protein is more conserved than the DNA. Okay, so protein is highly conserved than the DNA. That is why we take the protein as our comparison. Because if you look into the DNA, even whatever the speaker has presented in the morning, right, explained it. So glutamic acid to aspartic acid, if we take that as an example, at the DNA level, you will find it out as a mutation, right? Then you will mention it as a mutation that has happened. But at the protein level, change from aspartic acid to glutamic acid or glutamic acid to aspartic acid is not going to affect its functionality, because both are similar amino acids. So protein is not going to get affected with that mutation. That is why first is at the protein level, then look at the change in the amino acid that has happened. If the amino acid change has happened from one, let's say the hydrophobic to hydrophilic amino acid, or let's say the aspartic acid to isoleucine, if that mutation happens, then we know that this change is not allowed or is not going to affect the protein entirely. Please remember, no mutation is bad. Okay, mutation happens for its survival. Okay, even the coronavirus, the mutation is happening in the virus for its survival. Okay, it is just that it is creating a bad effect to the human, but the virus is undergoing the mutation for its survival. Even in the humans, the mutation is happening within us. It is happening for us to survive. Okay, so it is too difficult to make a conclusion that it's a bad mutation. Okay, because the mutation can be advantageous or disadvantageous to us, but it is for the finally the survival. So in that particular context, so if the look at the protein level mutation, and if the mutation is affecting its structure and stopping its functionality, then it is bad. Then go back, look at the DNA level, what is that change happened, then one can think of the gene editing or any other things. So that's how the analysis, that's how we should use this BLAST or FASTA to perform the experiments and then look at the results. Any doubts with whatever we have discussed?
Sir,
Yep.
Sir, at the start of the practical session, you told about the model organisms. How to select the model organisms? What are the criteria to select the model organisms?
See, the model organism, there are different, like E. coli, Drosophila, right? But it goes with whether the gene is present in that organism or not. Okay, so people are using even the elephants, horses, camel, okay, are used for testing in the clinical trials. We think always about the mouse or rat, as not the case. Clinical trials, in few cases, people use horses, elephants. Okay, so it just goes with if the disease is like leprosy, right? Armadillo is used as organism to test. So in that particular context, the gene should be there in that organism to express. So if it is there, we'll use it and then test it. But if the organism is too difficult to handle, okay, so we prefer to work with the smaller organisms. That's the major thing. But if the gene is not there in the smaller organisms like rat or mouse, then that's where the phylogenetic analysis comes into picture, that we will do in the next experiment, which is that next organism that we can handle. People do use monkey also, right? But it is slightly difficult to handle at the experimental setup. So maintaining those animals itself is a challenge. So model organism will vary. For humans, mouse is widely used because most of the genes that we have is there in the mouse. But the gene expression should also happen in that. What if by any of the methods, if it is not expressing in that, then we cannot use it as a model organism. But criteria is the gene and its expression. That gene should be there in that organism. In both the ways, we should be able to express it and we should be able to stop it as a negative control. Both should be there. Any doubts from anybody?
Sir, for humans and other animals or plants.
For the
Sir,
yeah.
For the multicellular organisms, yeah, what you are telling is right, sir. But we are talking about the microorganism like the coronavirus. They are also having some gene organism. And then what they are considered as a model organism, like we conclude that COVID is a new form of SARS virus, like that and all we are confining. And on what basis they are confining like that?
Yeah, good. Now, if we look into those viruses, right, coronavirus is not like a new virus. People like the coronavirus is there since decades, but the new variant that it is taking, right, that is making it. That's why I mentioned variant is nothing but the mutation that is happening. So the mutation that it is undertaking is making it as a pathogen. So every time it is changing. Viruses are self-replicating and their mutation rate is very high compared to any other organisms. So they will adapt to the required environment and goes for a change. And the new drug, whatever is made, it knows that if I make this change, the drug is not going to work. So that changes is going to happen in that virus. That made it very difficult. So what made it to have like that? Because for its survival. Okay, that's where finally it boils down to. Even we also change based on the environment, right? Our genome will go for a variation, environmental stress, but the genome level is constant, but the protein expression will keep varying within the humans. So that's how the evolution has happened from one to another one. So that's keep happening. Suddenly a person will change, right? So not at the genomic level, but at the phenotypic level. So making him a successful guy or into a miserable guy, both the ways. So if the coronavirus, frankly speaking, in our body, we have 100 plus of bacteria and viruses present in our body. Okay, it is not like we do not have any microorganisms being present. We have many microorganisms being present. They are all surviving, okay, because they know their limitations, and to survive they need a host, and the virus or bacteria are using our body as a host, and they are also living. Since they are not disturbing us, we are also asking them to live. Okay, both are there. Problem with the coronavirus is, if it has to survive, okay, it has to undergo a mutation. There is no other option. But the mutation should happen in such a way that it should allow us also to live. Then we would not have bothered about the coronavirus, because previously also we are affected with coronavirus. But the new variant that it is taking, it is killing the people, and it is also getting killed. So that is what it is not aware of. So the moment it gets to know that if I am not going to do this, and if I make myself these changes, right, it's something like you are killing others and you are also getting killed. So then you will realize that no, this is not the way. If I have to live, I should also make others to live. So that is what is taking the time. Then over a period of time, then it will realize, no, if this is what I need for the survival, and if I live like this in this environment, I will also survive, and the person is also going to survive. Then nobody think of making a drug molecule. That's how it is. We have bacteria, so many bacteria that are there. They are surviving by helping us in digestion. We are not killing them, right? We have good bacteria in them, in our body or in the gut. That's how it is.
Sir, if we are mostly the mutation, whatever happens, it will be the addition or deletion of DNA will occur to the next evolution or survival or the degradation, something to the organism. But the protein mutation doesn't allow us to do anything. It's just performing its action only. Then why we are giving importance to such protein today now?
Like, not understand the question.
If any mutation occurs in DNA, the organism, that particular organism will go to next evolution level. But the mutation in protein, I think that doesn't perform any functions related to that mutation. So why we are giving this much importance to protein to understand today's classes?
No, it is not like all the mutations are neutral in nature. Okay, if the proteins are undergoing mutation, but if the mutation is not going to affect, okay, any mutation is for the betterment of the protein. Okay, but some we may accept or may reflect. It's something like everybody work, but few work will get highlighted. It is not like nobody is going to work. All the members, everybody is going to work, and few will get highlighted, few will not. It's the same thing. So all the proteins in one or the way will go for a change, but few changes is only for its optimization. Okay, but few things, whatever the change happens, will make that protein to go to a next level. Okay, that makes either good or bad. Okay, both the things are there. So development is happening in the same way. Okay, so spike protein everybody is looking at, envelope protein everybody is looking at with reference to like coronavirus, right? Now envelope protein, there are non-structural proteins, because spike protein is the one which binds and then enters into. Is there a way by which we can make the spike protein not to act, or what are the mutations that is happening in the spike protein so that it can affect different individuals, right? What are the changes that is happening there in the genome? So if we can arrest the activity, then that protein will not be biologically active. So that is where the mutation analysis plays an important role. We really, in fact, in bioinformatics, we can also predict what kind of a mutation is going to happen in the future. okay, based on the complete background, analyzing the data, quantum of the data, frequency of the mutation from one amino acid to another amino acid. That's what is available in the plasma matrix, right? Somehow we can guess the kind of a next mutation that is going to happen. If it is -3, right, then if it is going in a virulent manner, then the chances is -4. If it is towards the less affection, effect is less, then -2, then -1. So we can go for a kind of a prediction. What is the next probable mutation that can happen? That's how the vaccine development or the prediction of the new variant people are making. It may happen, it may not happen, but it's a prediction. Okay. But yes, structural changes are more dangerous for the humans, but it's good for the virus to survive. It is for its survival that is happening.
Yes, sir. Now you are telling that, sir, that rat, something for a small organism, we are defining the large organism. For rat, we are telling humans, right? If some person suffers with the chromosome 6 disorders or some royal disease or any genetic disorders, that will not match the rat right now, sir.
Exactly, that's where I mentioned the gene, whatever is causing the disease, if that gene is present in that organism and can create the, see, in organisms, it is not going to have those diseases, okay, whatever we are facing. Now you need to induce the disease. So even if you want to test the HIV/AIDS, it is not like the HIV infection happens to the rat, but you got to induce the disease. Then, as I mentioned, leprosy and armadillo organism is not going to get affected with the leprosy, but you got to induce the disease, then make that organisms to express the disease, right, those limbs and other stuffs. Then you start giving your treatment method, then check whether it is getting cured or not. Once, otherwise, finding that organism where this disease is there, that's going to take huge amount of time. We may not able to get any drug or any vaccine at all. You may need to wait for centuries for the armadillo to get that leprosy, okay, because it is resistant for too many things. So many organisms are not having this coronavirus because they are resistant. It is not like they are not affected with coronavirus. This virus cannot survive in their gut at all or in the lungs at all. So not going to affect. But if there is any organism where we can induce the disease, then induce it. That's where people use the organisms. Right? So you got to induce and then check. Of course, in animal testing, there are different terms people use, like sacrificing. I don't know, the term itself is not right. There are many critics also available in the animal testing centers saying like, it is not like the person has sacrificed that organism for its survival. Okay, it is the organism which has sacrificed itself for the testing, right? In the wet lab, people do mention like, we sacrificed 20 organisms to test this particular method. No, you are not sacrificed. It is the organism which has sacrificed itself to test your hypothesis. So people are working on it to change those sentences, all the stuff. But nevertheless, identifying those organisms is not an easy, because we have thousands of organisms. Which one to use? What is the proof that it is going to express? So that is where the bioinformatics plays an important role, to test whether the gene is present or not. If it is there, possible to express our promoter region, all those things comes into picture later. How we can induce. Good. Any other questions?
Sir?
Yep.
Sir, I have a confusion, sir.
Yes.
What's the difference between a bioinformatic database and a tool, sir?
Bioinformatics database is like GenBank, okay, where you have the data present. SwissProt, GenBank, PDB, these are all your databases where the data is stored. Now the tools are BLAST. We are using the tools to extract the data. So those are the tools. So database is static, whatever is there in the behind. So whatever we see and give some instructions, right? So for example, whatever we did, BLAST, FASTA, these are the tools by which we can give our query like human CCR5 and ask the tool to search within the database, that is the GenBank or PDB or SwissProt Tremble, then give us the result back. So tools are our interfaces through which we will access the database.
Thank you, sir. I have another doubt. Is UniProt considered as a primary database, sir?
No, UniProt is a secondary database because
But we are collecting the data from it as well, so it's not considered as primary.
no, because the data for the UniProt comes from the GenBank.
Yeah.
Yeah. So that is why it has become like a secondary database. EMBL, GenBank, and DDBJ are the primary databases because we deposit the data to GenBank or DDBJ. We cannot deposit the data into the SwissProt. So that is why it becomes like a secondary database.
Okay, sir. Thank you.
Excellent.